Dear participants, I hope you can hear me. We will start soon. Welcome, first of all of you. We will start in a few minutes. And I would like to remind that the symposium will be broadcasted live on YouTube. And in 2 minutes, the presentations will start. We're just trying to give a little bit more time for all participants to be able to join the session. And I'm going to share the live stream in the chat box. If you would like to share it with your friends, your families, we would be very happy. I'll share the link in the chat box now. It should work.
Okay, dear participants, researchers, speakers, and guests, we would like to extend our warmest welcome to you all for the second annual Robert College High School Science Symposium.
Our intention in organizing this event has always been to promote scientific research among our peers and provide an environment in which they will have the opportunity to think and write about and discuss topics that will deepen their understanding of and interest in science. Therefore, the main philosophy of HiSci is not that of one competition. We rather hope to create a platform for high school students to share their passion for science and inspire one another.
We would like to start this event by thanking all authors and researchers that are participating in this year's conference for the time and effort they have put into crafting their research papers. The effort that our researchers put into their work was quite appreciable and inspiring. We also would like to thank the faculty, both from and outside Robert College, that have assisted us in the evaluation process. This symposium would not be possible without your valuable contributions. On behalf of our dear evaluators, Zeynep Atay, Ece Kara PhD, Dünya Önen, James Butterworth, Hazal Özülhan, Klaus Kadoret, Professor Fatih Uğurda PhD, Professor Sezer Gören Uğurda PhD, Sean van der Meulen, and Professor Beren Semiz Gürsoy PhD, we will plant 35 trees through the Tema Foundation. We hope that you accept this donation as a token of our gratitude.
Before we move on, we would like to recognize the participating schools for promoting our mission of spreading knowledge and inspiring one another. İzmir American Collegiate Institute, SEV American College, Koç School, Darüşşafaka Schools, Robert College, İstek Acıbadem Anatolian High School, Rutgers Preparatory School, Crystal Springs Upland School, St. Mark School, Palmer Trinity School, Başkent University Private Ayşe Abla School, Ankara Çözüm College High School, Saint Michel French High School, Trinity Valley High School, Brewster Academy, Ankara Science High School, Private Bahçeşehir Anatolian High School. We would like to thank you, both the students who have taken the time to send papers and participate in this event, and the advisors who have guided young researchers in this process.
Now, we'll briefly go over the schedule for today's symposium. After the opening session, we will invite the author of one of the papers to deliver their presentation. Then, we will have a 5-minute break, following which we will invite our keynote speaker, Professor Dr. Batu Erman, to deliver his speech on genome engineering. The presentation will be followed by a 5-minute break, after which we will invite the authors of 2 more papers to deliver their presentations lasting 20 minutes per paper, with a 5-minute break in between the 2 presentations. Afterwards, Professor Dr. Naz Zeynep Ataygök, our second keynote speaker for this year and the head of the Department of Chemistry at Boğaziçi University, will deliver her speech.
At 7 Istanbul time and 12 p.m. Eastern time, we will have an hour-long break. After the break, we'll proceed with the presentations of the remaining 3 papers. Again, 20 minutes each, with brief breaks of about 5 minutes in between them. After the presentations, we will conclude with the closing session, in which the prizes will be announced. That being said, we will be yielding the floor to our fellow presenters, Akif and Ipek.
We will now proceed with our first paper presentation. Punavit Kasian from Brewster Academy in New Hampshire will now present their research, finding the exact value of cosine of pi over 7. We would kindly request our participants, including those following us on YouTube, to take note of their questions. There will be time after the presentation for them to ask questions. Punavit, you now have the floor.
Thank you. Do you hear me clearly?
Yes, we do.
All right. Good morning, good afternoon, and good evening, people around the world. So I have to share my screen, right? Share my presentation.
Yes, you can share your screen now.
All right. All right. Good morning, good afternoon, and good evening, people around the world. My name is Punavit Kasian. I'm going to present you my math research, finding the exact value of cosine pi over 7. Let's begin. Here are 4 keywords in my work: trigonometric functions, cubic equation, Cardan's method, the method to solve any cubic equations, and complex plane, refer to a plane where the horizontal axis is the real part of imaginary numbers, and the vertical axis is the imaginary part of the imaginary numbers. So next, the introduction. The values of trigonometric functions at pi over 6, pi over 5, pi over 4, and pi over 3 have simple root expressions. It isn't easy to find a record on the values of trigonometric functions at pi over 7. An article from Wolfram Medward states that cosine pi over 7 is a solution of a cubic equation, and sine pi over 7 is a solution of a cyclic equation without giving root expressions of both quantities. It is easier to solve the cubic equation for cosine pi over 7 and use a trigonometric identity psi square x plus cosine square x equals 1 to find sine pi over 7. We will move on to the objective. This research has two objectives. Firstly, to solve the equation stated in the Wolfram-Medvold article is correct. Secondly, to find the exact value of cosine pi over 7, cosine 3 pi over 7, and cosine 5 pi over 7. At this point, some of you may wonder how cosine 3 pi over 7 and cosine 5 pi over 7 are involved, since I started talking only about cosine of pi over 7. You remember the cubic equation to solve for the cosine pi over 7, right? My further investigation revealed that the other two answers to the cubic equation are cosine 3 pi over 7 and cosine 5 pi over 7, which you will see later in my presentation. Let's move on to the methods. First of all, I studied how to create an equation and then solve for sine pi over 10. After that, I verified that cosine pi over 7, cosine 3 pi over 7, and cosine 5 pi over 7 are the solutions to this cubic equation. Later on, I solved this cubic equation using Cardan's method. Lastly, I identified the root expressions with cosine pi over 7, cosine 3 pi over 7, and cosine 5 pi over 7. Now we are in the main part of this presentation, result and discussion. Since this section contains much information, I would like to divide this section into 3 parts, just like the methods. To begin with, I will prove the equation stated in the Wolfram-Medvold article. Then we will see how I solve this equation using Cardan's method. Finally, I will identify root expressions with cosine pi over 7, cosine 3 pi over 7, and cosine 5 pi over 7. To show that the cubic equation is true, I began by defining that A equals pi over 7. Now we get that pi minus 3a equals 4a. I take cosine function to the both sides and get that negative cosine 3a equals cosine 4a. I will stop right here first to show that in cases of 3 pi over 7 and 5 pi over 7 are the same as pi over 7. So similar to pi over 7. Now I define that b equals 3 pi over 7. Now we get that 3 pi minus 3b equals 4b. Again, I take cosine functions to the both sides. And I know that negative cosine 3b equals cosine 4b. As you can see that the equation is exactly the same as in case of pi over 7. It will also be the same equation for 5 pi over 7. Now we know that in case of 3 angles lead to the same equation by applying some trigonometric identities to the above equation. Now we receive the trigonometric equation in terms of only cosine function and angle A. Next, I factor the equation. We all know that cosine pi over 7, cosine 3 pi over 7, or cosine 5 pi over 7 is not equal to -1. So we can ignore the first factor in front, cosine A plus 1. Now we can prove that the cubic equation is true. Moving forward, I'm going to show you the Cardan's method at the moment. As aforementioned, Cardan's method can be used to solve any cubic equations. There are a few steps. Firstly, I substitute x equals t minus b over 3a. We know that p of t minus b over 3a equals 3q, t cubed plus pt plus q. Afterward, I identify that t equals u plus v is the root of polynomial t cubed plus pt plus q. Then we get that u cubed and v cubed are roots of a polynomial y squared plus qy minus p cubed over 27. Next, we solve for u and v from the polynomial I just mentioned and the equation uv equals negative p over 3. Lastly, the solution to p of x is solution to p of x equals 0 is x equals u plus v minus b over 3a. After applying Cardan's method to the cubic equation, I received these three answers. Now, I'm going to illustrate how to identify root expressions with cosine pi over 7, cosine 3 pi over 7, and cosine 5 pi over 7. So I consider cube root of 1 plus 3 square root of 3i. Then I pull out the cube root of 2 times square root of 7 from the expression. And we will consider the cube root of the imaginary number that lies on the first quadrant of the complex plane only. Now we are considering the real part of two imaginary numbers. The real part of z1 is less than of z2. So hence, we get that the real part of z2 is greater than one number. When we plug in that number to the expression, we now know that one answer is negative. Therefore, right now we know that the value of cosine 5 pi over 7, since it is the only value that is negative. Further analysis in the complex plane, we can compare the other two quantities. Thus, now I can identify all of the answers because we know that cosine pi over 7 is greater than cosine 3 pi over 7. Also from the complex plane, we can rewrite the expression in the simpler forms as shown. RE of z is the real part of an imaginary number z. Because we know that the value of cosine 5 pi over 7, we can apply a trigonometric identity to obtain the values of cosine pi over 7 and cosine 3 pi over 7 that do not involve omega in the expression. Moving on to the discussion. The summation and product of roots are equal to 1/2 and -1/8, respectively, which correspond to the relation of roots and coefficients. We have come to the last part of the work, the conclusion. Here are the exact values of cosine pi/7 and cosine 3pi/7 and cosine 5pi/7. For the acknowledgement, I would like to express my very great appreciation to the to the promotion of teaching science and technology IPST and the development of promotion of science and technology talents project DPST in Thailand for project support and funding in 2020. I would also like to offer my special thanks to Dr. Nantawat Udom-Chabitak from Mahidol University for his valuable support and constructive suggestions, advising during the planning and development of this project, and mentoring me for the presentation. Moreover, I rewrote this research paper in 2022 while attending Business Academy. I must also thank Dr. Ben Vendel from Business Academy for rechecking this paper in terms of citation and references. Here are my references. Thank you for your attention. I'm happy to answer any questions.
Thank you. It was a great presentation. We will now proceed with the Q&A session. Participants may ask their questions verbally by raising their hands or send their questions as messages through the Zoom chat, or participants who are viewing this event from YouTube may use the chat function to state their questions.
Yes, I see some hands.
Yeah, I was going to ask, while preparing this report, were the practical applications of this topic were more important to you, or the theory behind the solution?
So sorry, can you say that again, please?
Am I audible?
Your question.
Okay, I was asking, while preparing this report, were the practical applications of the topic was more important to you, or the theory behind the solution?
You mean the application or the so I'm sorry.
I was just asking, like, were the practical applications of this concept in real life cases or in mathematical problems were more important to you, or rather than the theory behind the solution part?
I see. So the application of this research, actually, like I don't know how to use it in real life, but it will expand the knowledge in trigonometry and in math. Does that answer your question?
Yeah, that answers it. Thank you a lot.
I have a question. Other than the cosine of pi/7, are there any other trigonometric expressions that can be found by similar methods?
Is that what?
Like other trigonometric expressions that can be found by similar methods, other than the pi/7.
So the trigonometry of function at pi/7.
Yeah, other than that.
If you're having difficulty understanding it, maybe Tunahan could write the question through chat.
Oh,
Would that be okay?
Yes.
I will wait for him to write the question. In the meanwhile, Salim.
First of all, hello again. Thanks for the presentation. It was a delightful one. I wanted to ask, when we saw the outline, I first had the question, how does one know to intuitively start from like the cubic shape equation? And because you know, yeah, you start from define px equals that equation, and how does one know that intuitively that's the starting point, and then you go onward from that solving the equation?
So I okay. You mean the Kadam's method, right? The method that I use to solve the cubic equation, right? Like this method can be used to solve any cubic equation. Like for some of cubic equations, you cannot just factor it. It's not that simple. So we have to find another way to solve it, to solve any cubic equations. Does that answer your question?
Yes, thank you. You're
I think we also have a question in the chat, so I'm going to read it for everyone. Other than the cosine of pi over 7, are there any other trigonometric expressions that can be found by similar methods?
Similar method. I'm yeah, I think it can be solved by similar methods, as like you for example, that I have an idea from psi pi over 10 from creating an equation and solve an equation. I think it's like this for all the values, whatever the angle is. You can so first thing you have to do is to find the equation first and then solve it. But the difference is that for some expressions, like mine is cubic equation has three answers. So I have to identify which answer is which one. Which answer is cosine pi over 7, which answer is cosine 3 pi over 7, and which answer is cosine 5 pi over 7. So the identifying roots expressions will be different.
Thank you very much. Okay, is there another question? Yes, Ekin.
Okay, thanks for the presentation. So what is the significance of knowing the exact value of cosine pi over 7 in mathematical practices?
Oh, you mean the application? Like as I mentioned before, that I don't know how to use it in real life, but it will expand the knowledge of trigonometry and mathematics.
Yeah, thank you very much. That was the time we had for questions. It was an amazing presentation. At 45 past, which is 8 minutes later, we will be inviting Professor Erman. The participants can now turn off their cameras and microphones for a short break, but please don't leave the Zoom meeting. For those who are attending to Zoom right now, we're giving a very short break before Dr. Arman gives his speech. Because of our schedule, we're a little bit ahead of our schedule. We would like to wait for the right time for Dr. Arman's speech because there will be other people probably attending on the right time. Thank you for your understanding, everyone. We will start shortly. You can have a short break at the moment.
Hello, everyone. So that was the end of the break. We kindly request that participants turn on their cameras. Before we proceed with the keynote speech, we would like to introduce our keynote speaker, Professor Erman.
Dr. Batu Erman is a professor in Boğaziçi University's Molecular Biology and Genetics Department. Dr. Erman graduated from Robert College in 1987, received his bachelor's degree in Hamilton College in 1991, and his PhD in biology from Brandeis University in 1998 due to his research about regulation of immunoglobulin heavy chain transcription. He was the first Turkish scientist to receive the prestigious Marie Curie Excellence Award. He was also a postdoctoral research fellow in Dr. Alfred Singer's laboratory at the Experimental Immunology branch of the National Cancer Institute and National Institutes of Health from 1998 to 2004. Upon his return to Turkey, Dr. Erman joined the faculty of Sabancı University in 2004, and since 2015 is a professor of molecular biology, genetics, and bioengineering. He consequently moved his lab to Boğaziçi University in 2020. Before yielding the floor to Professor Erman, we would like to thank him for accepting our invitation. We would kindly request our participants to take note of their questions. There will be a Q&A session at the end. Professor Erman, you now have the floor.
No, you can't now. I hope you can hear me now. Is the sound okay? Good.
Yes.
So thank you very much for this invitation. I hope I can talk a little bit about genome engineering, which is a very new subject, but at the basis of it is the kind of molecular biology and genetics experiments that we do in my lab. So what I plan to do today is to give you a history of a brief history of the genome engineering field, and then show you some experiments that we've done using these tools, CRISPR-Cas9 tools. So let me share my screen, and hopefully you can see full screen. Yes. Unfortunately, I don't see any of you, so you have to speak up if something goes wrong. But let me just. Yeah. Okay. Okay. So today I'll talk about genome engineering, and this is not your traditional type of engineering like chemical engineering or electrical engineering, but the kind of things that you can do as a molecular biologist or a cell biologist or maybe an immunologist like I am. And at the heart of it lies, of course, the central dogma of Watson and Crick. And you know probably this central dogma. It says that all genetic material is hidden in nucleic acids. They could be DNA or they could be RNA. And then this information is passed on to messenger RNA. And then from messenger RNA, it gets turned into proteins. And the proteins are the working workhorses of the cell or the virus. And they have structural functions, or they can have enzymatic functions, or they can have signal transduction functions. There are many different types of proteins. But basically, the central dogma says the genotype, the genes that are in the sequence of DNA or RNA molecules, it controls the phenotype, which is how the proteins behave. And the field of genome engineering is the ability to change the genes, to change the genotype, and to alter or change the way a cell behaves, or to generate new functions and create new structures or new enzymatic reactions or new biological processes. So that is the traditional engineering. You design and you apply your design to some structure, and you hope that it behaves in the way you designed it. And if it doesn't, then you change your design slightly, and then you try again. This is exactly what civil engineers do when they design bridges to cross rivers. Of course, there's much more design in traditional engineering because you can't have it fail. But as an experimental genome engineer, you can try and perhaps make some mistakes, but in the end, you build on your mistakes. So if you have any questions about what I'll talk about, you can email me at this email address, or you can Google the type of our website and learn more about the kinds of research that we do. So what I would like to start this talk with is a discussion of this Nobel Prize in Chemistry in 2020, so 2 years ago. And this award was given in 2020 to two women, Emmanuelle Charpentier and Jennifer Doudna. And this was for the development of a method of genome editing. So I use the word genome engineering and genome editing interchangeably. And but they were, so this was a big breakthrough, and the discovery and the Nobel Prize, the amount of time it took it was quite fast. So it's so groundbreaking, almost as significant to biology as Watson and Crick's discovery of the double helix structure of the DNA. So there was a surprise also when these two ladies won the Nobel Prize, because there were some people who did not get awarded, and there were some controversies. But I would like to point out something. So, for example, when you win a Nobel Prize, if you're at that stage of your career, typically you don't just wake up one morning and there's a phone call from Sweden and you're pleasantly surprised that you won the Nobel Prize. Typically there are precursors to the Nobel Prize. So, for example, the Lasker Prize in Medicine is a prize in the past 121 people who won the Lasker Prize. Almost half of them went on to win the Nobel Prize. So most likely, if you win the Nobel Prize, you're also awarded previously the Lasker Prize. And there's another prize called the Kavli Prize, which actually pays much more money than the Nobel Prize. And in fact, in 2018, Emmanuelle Charpentier and Jennifer Doudna also won this Kavli Prize together with a scientist from Lithuania. But again, what was missing was this other person, and this is the controversy in this Nobel Prize. Feng Zhang, who was a professor, or I guess still is a professor at MIT, was not among the people who won the Nobel Prize. So why is that? There were many arguments, but Feng Zhang actually, let me just tell you a little bit about him. This is a slightly outdated slide, but Feng Zhang works at this Broad Institute, and he was instrumental in making this CRISPR technique very popular, and allowed the distribution of the types of plasmids that encode the proteins that molecular biologists can use to perform genome editing using CRISPR-Cas9. So they used this company called AdGene, and from AdGene they distributed in 2020 only almost 60,000 plasmid copies of the genes that allow you to do CRISPR-Cas9 genome editing. And more or less all over the world, people have received these reagents. And today, many, including mine, almost all, let's say, molecular biology labs in the world use genome editing, use CRISPR-Cas9 to change the DNA sequence of organisms, model organisms such as mice or fruit flies or zebrafish, or tissue culture cell lines or cancer cells, and in some cases, as we shall see, in human patients also. Clinical trials that target certain diseases, genetically encoded diseases, and there are trials that try to change the human genome to try to alter the disease phenotype. Of course, there's the whole field of agriculture and veterinary science that this technique also is very amenable to. And in fact, not only did these scientists win the Nobel Prize and these other kinds of prizes, but they all have companies that have patents that protect the technology, so that if you want to do something, use this technology of CRISPR-Cas9 to develop something that you would like to sell, so you'd like to market it, then you need to buy a license from these patents of the companies of the scientists. If you want to use this for, like we do, for basic research methods, then you don't need to worry about patent protection or licensing. But this slide from a review, recent review, really shows how complex the situation is, because depending on the application, different institutes and different scientists, even though Feng Zhang and Emmanuel Champertier and Jennifer Doudna were the original discoverers of this, different institutes hold patents protect different applications of the technology. So, for example, if you want to use CRISPR-Cas9 in livestock, so you want to change the genome of a cow or a sheep, then you have to get a license from Broad Institute. On the other hand, if you want to change the genome of stem cells, then you have to get a license from Emmanuel Charpentier's company. Or if you want to use it for more medical research, then you have to get a different license. So it's a complicated field. Immediately after its discovery, it became this very complicated patent-protected field for applications. So it has a lot of applications. But to understand these applications, I think we need to talk about the mechanism. How does it work? And what can we do with it? And again, just like the Nobel Prize doesn't just appear one morning with a phone call from Sweden, earlier there was another Nobel Prize that was awarded to Capecchi, Evans, and Smithies, and this was for discovering principles of introducing specific gene modifications in mice by the use of embryonic stem cells. So I would like to talk about this earlier Nobel Prize and how it set the stage for the CRISPR-Cas9 patent and the Nobel Prize. So what Smithies and Capecchi did was they devised a method to change the genome of a living mouse. And we know, of course, that mice and humans and most mammals, were generated from an embryo, a zygote, a fertilized egg, fertilized by a sperm. And this egg, when it starts to develop in the womb of the mother, turns into, after a couple of cell divisions, turns into a blastocyst. And this blastocyst has this two types of structures. There's the structure that's on the outside, which is called the tropoectoderm. And then there's this inner cell mass, which contains the stem cells. These are embryonic stem cells, or ES cells. And you can actually collect these blastocysts from a pregnant female mouse and just scrape out all the embryonic stem cells, be careful not to kill the cells, and then start culturing them in a petri dish like you see here. And you could also genetically modify them. So you could take one cell and modify it so that you insert or you change some gene sequence in it and select this genetically modified, shown in pink here, this genetically modified cell, so that you only grow the ones that are genetically modified and you kill the ones that do not get the genetic modification. So this earlier Nobel Prize discovered how this could be done. So you could see that in a cell, you could target the cell, the genome of the cell inside the nucleus of the cell, using a resistance gene, an antibiotic resistance gene, that is placed inside of this cassette that has some DNA sequence that is homologous or very similar to the target gene that you want to target. So you could insert genes like this neomycin resistant gene into the chromosome in a single embryonic stem cell. And then once you select this embryonic stem cell, genetically modified embryonic stem cell, then you can, because embryonic stem cells have this property of self-replication and renewal, they and not only do they replicate and self-renew, but they can also give rise to all the cells into the body. So once you get this colony of embryonic stem cells that are genetically modified, then you can take another blastocyst and inject these genetically modified cells and then make a new embryo that has the genes that you inserted. So one component of this Nobel Prize was Capecchi's pipette injector, and you see a picture under a microscope of how this works. So this is a fertilized embryo before the blastocyst stage, and here's a pipette that's sucking the embryo to a specific point under the microscope, and you can actually see inside the male and the female nucleus. And with a separate pipette, you can inject DNA, or you can just pull out the whole nucleus and replace another nucleus, and this led to the concept of cloning animals. And here you see using a similar pipette. The pipette on the left is holding a blastocyst, and here you see the trochoectoderm on the outside, and here is the inner cell mass. And with this pipette from the right, you can insert these or inject the genetically modified cells into the inner cell mass. And you see here, these are time course experiment, and you can see these cells are injected into the inner cell mass. So these would be the genetically modified cells that repopulate this inner cell mass. And if you could take this blastocyst and implant it into a female, then the embryo that this blastocyst gives rise to would be genetically modified. So this one was the technology in the early, between the 1980s and the early 2000s that allowed cloning and genetic modification of animal models. But this was a very inefficient process. So you have to generate a lot of blastocyst injections to get the one that you want. So now we come to CRISPR-Cas9. How does CRISPR-Cas9 make this much easier so that every lab in the on the planet uses it? Well, what it does is actually it generates an enzyme that allows cutting of first the recognition of a DNA sequence and cutting of this DNA sequence. So here is an enzyme in blue that binds to specifically recognizes a DNA sequence. And here is an enzyme that is a nuclease that makes breaks in the DNA sequence. This is what CRISPR-Cas9 actually is. Now we shall see that it's not that simple. It's not only one protein binding to DNA and cutting DNA. It's a little bit more complicated than that. But the idea of having proteins that recognize DNA or proteins that cut DNA, this is not a new idea, because we know that there are these transcription factors that control all the transcription events. So the reading of the genes and turning them into mRNA, these are controlled by transcription factors. And in fact, transcription factors are these proteins that really don't have any enzymatic activity, but they have the ability to bind to DNA. And they have, so they have this DNA binding domain. I called it the DBD. And they also have activation domains through which they interact with the RNA polymerase machinery that allows transcription, so generation of messenger RNAs. So transcription factors are very, very important. We study them, and I'll show you some examples later on. But this lies at the heart of designing or engineering this kind of enzyme that would bind to DNA and also cut DNA. So normally a transcription factor using its activation domain would recruit RNA polymerase to the start of the gene, and that would cause gene transcription or generation of messenger RNA. The second type of protein that we already knew from the early 1970s are restriction enzymes. And we use restriction enzymes to cut DNA. In fact, to do cloning, gene cloning, we use this restriction enzymes almost every day in molecular biology labs. And what these do is they recognize a sequence of DNA and they make a double-stranded break, or they break both strands of the DNA, double-stranded DNA molecule. So what is needed then is a hybrid enzyme that has the ability to bind to a sequence-specific DNA molecule and also has the ability to make a double-stranded break in the DNA molecule. So it's a fusion, a hybrid between transcription factors and restriction enzymes. In fact, before we came to CRISPR-Cas9 in the early 1990s, so 1996 is this paper, several papers, people were working on this to design these kinds of hybrid restriction enzymes. What they did is they used DNA binding domains of transcription factors called zinc finger transcription factors that recognize a certain sequence of DNA and stick to it, and also a restrictions enzyme called FOC1. And FOC1, what it does is once it comes close to the DNA, it makes a break on the top strand and another break on the bottom strand of the DNA. So these enzymes, actually, they were elected the method of the year by Nature journal, which is a well-respected science journal. In 2011, it was the method of the year, and these are called zinc finger nucleases. And but there were some problems designing this DNA binding domain so that it has to bind only a certain sequence was not that straightforward. But at the so it didn't really catch on and become so popular as CRISPR-Cas9. And I'm talking about the history of this to try to emphasize why now it's so popular, CRISPR-Cas9. So these zinc finger nucleases, they recognize DNA using this zinc finger motif. And the zinc finger motif comes from these transcription factors. And in this slide from this paper, you see the different types of transcription factor proteins that are encoded by different genomes. So in blue, you see Saccharomyces cerevisiae, the yeast, which is a model organism. In yellow, you see the C. elegans. In red, you see Drosophila melanogaster. This is a fruit fly. And in green is the human genome. And these are the different families of transcription factors. And regardless of the genome, what you see is the majority of the transcription factors use this zinc finger motif to bind to DNA. So it was really straightforward to take this zinc finger motif as the DNA recognition component of these hybrid enzymes. So you see that in humans, in the human genome, there's almost 1,000 transcription factors. So the whole human genome is roughly 20,000 genes. Of those, there is about 5,000 transcription factors, but of those, about 1,000 of them use this zinc finger motif to bind to DNA. So it's very, in evolutionary terms, it's very popular to use zinc fingers motifs to bind to DNA in a sequence-specific way. So this is how these zinc finger nucleases worked. They're called zinc fingers because the structure contains this little finger, and that's coordinated by a zinc atom shown in this red circle. And it recognizes 4 bases, 3 bases on the top strand of the DNA, 1 base on the bottom strand of the DNA. And if you have 4 zinc fingers, for example, then you can recognize up to 12 nucleotides in the DNA sequence. And using this linker, you can fuse a restriction enzyme that would cut the DNA. And you can have 2 zinc fingers, one binding to the right, one binding to the left. And this FOC1 would cut once on the top strand and once on the bottom strand and generate this double-stranded break. So this was in the 90s. This was the way to do genome engineering. It never picked up. I mean, there are some companies that made millions and millions of dollars using this technology, and many labs used it, but it was not as popular as CRISPR-Cas9. So then other scientists discovered another method of cutting DNA, and these are called talons. And the talons were really interesting. My lab worked on it for about 5 years. And the talons were derived from this type of bacteria called Xanthomonas. And the Xanthomonas in nature is a plant pathogenic bacterium. So here you see a picture, I picked it up from Wikipedia, and this is a plant leaf that has been infected by this Xanthomonas bacterium. And what the Xanthomonas does, it has the bacterial cell has this nano size injector through which it injects its own transcription factor into the plant cell. So the injector goes into the plant cell, passes through the wall, cell membrane, and the transcription factor made by the bacterium goes and binds to the genome of the infected plant cell. And it binds to genes that would regulate sugar transport. And that way, the plant cell starts to exude glucose, and the bacterium that's on the outside of the cell of the plant will start to eat this glucose. It's an evolutionary adaptation mechanism. Now, what you see is that the plants have this immune response against this bacterial infection, which is that at the site of infection, there's this local necrosis. Necrosis is a way of cell death. So these plant cells die when they're infected so that the bacterial infection does not spread systemically to the rest of the plant. So there's this give and take, or this competition between the bacteria using the plant's genes to make sugar, and the plants trying to avoid the infection by committing suicide locally in a cellular context. Okay, so what these talons look like was this really pretty picture. This is a crystal structure. X-ray crystallography experiment showed us this crystal structure. And the two things show the same thing. So here in the middle, you see the DNA double helix in gray. You see the ladder going up. And here we're looking at the DNA from the end. So the phosphate backbones are on the outside and the bases are on the inside. And around the DNA helix is an extra helix, which is these xanthomonas talon protein. And each color represents a single finger that gets inserted into the DNA. You can actually see. So, for example, here, one finger gets inserted here in green, and it contacts the two bases in the middle of the DNA. So it's a way in evolution how these bacteria have discovered a way to bind to DNA and recognize only a certain sequence of DNA. So these are these xanthomonas tail proteins, and the tail stands for transcription activator-like effector. Complicated long name, but what it is it's a transcription factor. It has a DNA binding domain that has these repeats, and it has an activation domain that talks to the RNA polymerase machinery to activate the gene transcription. And you see here that there are 18 repeats. Each repeat has 34 amino acids. I've colored them in the colors of the rainbow. Each repeat has exactly the same amino acid sequence. You see it here, 34 amino acids, except for the two in the middle here. And there are 4 types, and those 4 types recognize the 4 bases of the DNA. So there was this code for the tail code that you could use. You could look at the sequence of the protein, and looking at the repetition, and you can look at what these 2 amino acids are, you could figure out which DNA sequence it will bind to in the genome. And once you figure out the code, then you can mix and match and change the repeats. And when you change the repeats, then you can alter the DNA binding specificity. So these were done. And moreover, what you can do is you can get rid of the activation domain. So it no longer communicates with the RNA polymerase, but you can put this FOC1 restriction domain. So the talon protein would bind to DNA using its DNA binding domains, and then it would make a double-stranded break or cut the DNA in the middle. Okay, so these were early attempts at genome engineering. And here I show that. So you have a talon protein. There's a right protein and a left protein. They bind to 2 different sequences, and the scissor symbolizes the enzyme that would cut the DNA. So why do we want to cut the DNA? In fact, if you want to engineer DNA, you can change the DNA. You don't want to really cut it. You want to change it, right? You want to change the sequence of the DNA. Well, it turns out that when you make a double-stranded break, most cells have a response, a DNA repair response, because cells do not like breaks in the middle of chromosomes. So immediately they try to repair the break. And there are 2 ways of repairing this DNA. One of them is called non-homologous end joining, NHEJ. And this is an error-prone mechanism by which DNA can be repaired. So once the break is done, then you generate indels or insertions or deletions. So you make mistakes. And the second way, mistakes means mutations, and the more you cut the DNA, the more changes you make, but the changes are random. The more sort of precise way of modifying the DNA is after you cut, you can use homologous recombination. And cells, of course, we know our cells are diploid because if there's a problem with one of the chromosomes, the second chromosome can be used to repair the first chromosome. So if you make a DNA break in one chromosome, then the other chromosome sequence can be used to repair the DNA. So this is how we came to this CRISPR technology. So there were the precursors, earlier enzymes that could protein-based enzymes that could cut DNA and cause these mutations. Now, when we come to CRISPR, of course, as you probably all know, is not only a protein enzyme, but it's a combination between an RNA molecule and a DNA molecule. RNA molecule and a protein molecule enzyme. So here you see the DNA molecule that's going to be cut, and here is an RNA molecule, and in the ribbons is the protein. So this is the Cas9 protein. And this has been derived from nature as well, like many engineering feats have been derived from nature. And the Cas9 system, or the CRISPR system, many bacteria use this system to protect themselves from bacteriophage infection. So bacteriophages are these viruses of bacteria, and they inject their genome into the bacterium. And once that happens, the bacteriophage has these enzymes that chop up the incoming bacteriophage genome, and it places it into this chromosome of the bacterium that results in these short RNA copies of the sequences of the bacteriophage. And these sequences, these RNAs, they're called cRNAs. They are used by other enzymes to detect the presence of the DNA if it happens a second time. So this is a sort of bacteriological immunological memory. So you get infected once, you retain the information of the initial infection, and then if you get infected again, then the incoming DNA, instead of being inserted into the genome, will immediately be chopped up by these small RNA molecules, RNA protein complex. So this is exactly how CRISPR-Cas9 works. There's an RNA component shown in these nucleotide letters, and there's a Cas9 nuclease enzyme, which makes breaks in the DNA. So we call this RNA component the guide RNA, and the sequence, the first 20 nucleotides of the guide RNA have to match with the target DNA sequence. And once that happens, the Cas9 enzyme binds to this guide RNA, and it makes this double-stranded break. And as I told you earlier, when you make, regardless of what technology you use, whether you use Cas9 or talons or zinc finger nucleases, when you make a double-stranded break, the cell would like to repair that by NHEJ or by homologous recombination. So using this technology, you can use homologous sequences around the cut site to insert or to modify DNA sequences. This is the CRISPR revolution. There are, of course, many alternative ways of doing this. Instead of using the Cas9 protein, you can use a derivative of that, which is called a Cas9 nickase, which again binds to this guide RNA you see here in blue and red. This is the RNA molecule. Here is in the middle is the target double-stranded DNA. But because it's a nickase, it's not a mutant version of the Cas9. It doesn't make double-stranded breaks, but it makes a single-stranded break, only the top strand. Well, you can design, of course, two Cas9s, one binding here and a second one binding here, and you can make a cut here and a cut here, one on the top strand, one on the bottom strand, and again, still get the same effect of this non-homologous end joining or homologous recombination methods of repairing the DNA. So to get to CRISPR, people historically tried other enzymes to cut the DNA. What we're really trying to do is, this original Nobel Prize that Capecchi, Evans, and was won for modifying the genome of embryonic stem cells, that was relying on homologous recombination inside the embryonic stem cells. But now you can, with these enzymes, you can make the process much, much more efficient. And in any type of cell, not just only embryonic stem cells, You can cut DNA and during the repair make mistakes or make mutations or insert DNA sequences in any living cell. And once you have this ability, then you can use it for, as I told you earlier, veterinary science. You can change the genome of animals or plants. And in human patients, there are now clinical trials. So, for example, this company uses zinc finger nucleases, this earlier technology, to treat hemophilia. There's another company that tries to make change and repair mutations that cause this disease called beta thalassemia. And this company called Sanofi also is trying to design a drug that targets sickle cell anemia. And there are many genetic diseases that one can target. Of course, one of the limitations is that you can target one cell at a time. You can't target the whole genome. If you want to target the whole genome, then you have to target the embryonic stem cells. Or, for example, you could target all blood cells by targeting hematopoietic stem cells, which are the stem cells of the blood. So in the last 2 minutes, let me quickly tell you about one experiment that we've done using this technology in my own lab at OSG. So we're interested in this gene called POTS1, and this is a gene that's important for cancer. In many cancer patients, this gene is mutated or is highly expressed or is broken and made into fusions with other genes that drives the cancer to grow uncontrollably. So we wanted to understand what would happen if we remove this gene, or mutate the gene. So we designed a CRISPR, you see on the left, that targets just this gene at the beginning of the gene. And once we do that, then the cell, we make a double-stranded break, cutting once on the top, once on the bottom. And the cell that we do this in, so of course we have to insert the Cas9 protein and the guide RNA into the target cell. And we use cancer cells to do this that are indefinitely growing in the laboratory in tissue culture. And once we make the break, what happens is the gene is mutated during the repair and the cells lose the expression of the protein. So on the left here, I'm showing you an experiment called the Western blot. A Western blot is looking for the presence of proteins. And on the left lane, you see the present in the wild type cells, the wild type cancer cells. This protein is present. And in the cell here, the protein is absent. So we're detecting the presence of the protein using some antibodies that are specific against it. There are some other proteins that are around for controls, but here, if you look at the labeled band, it's absent. So once we target the genome, we change the genotype, we can change the phenotype, we can get rid of the protein. So that we call this making a knockout. So we knocked out the gene and there's no more protein. We can use other techniques like flow cytometry. This is a machine that looks for fluorescence, and you try to detect the presence of the protein using an antibody that is fluorescently labeled. And you see here in the wild type cells, all the cells are expressing the protein. They're sort of high on the x-axis. And in the mutant cells, they've decreased their expression. They've shifted towards the left, I'm sorry. So that these two techniques tell us that our mutation by CRISPR-Cas9 actually gets rid of the protein. Now the problem is what happens when we get rid of the protein? We don't really know. We need to find out what is the change in the phenotype. So to do that, we use a technique called RNA sequencing. This is a very sort of high-tech method of looking at all the presence of all the genes that are being transcribed. So what we're doing is we collect all the RNAs from our wild type cells. And in this case, we're using the cell line, which is a colon cancer cell line. And then these are the derivatives of the cell line that is a knockout for our gene. So we collect all the RNAs from the wild type cell, all the RNAs from the mutant cell, and we sequence all of those genes. And then we compare them, which one is elevated, which one is decreased. And what we see is there are 156 genes that are differentially expressed. So DEG stands for differentially expressed genes. You remove POTS1, 156 genes, their transcription levels change. 67 of them get upregulated when POTS1 is knocked out, and 89 of them are downregulated when POTS1 is knocked out. So that means that this POTS1, the protein that we're working with, is controlling these genes and somehow is regulating their expression. So we use some other techniques to show that. So what we find is that the genes that are in this group, there's some candidate genes that are important for cancer, and this way we can understand how our gene and the genes that it controls can regulate cancer development by making knockouts and removing genes, using genome editing, using the CRISPR-Cas9 technology. And you can do this for every single one of the 20,000 genes in the human genome and look for the phenotype differences, the phenotypic effects. And these are being done in all labs around the world. And you don't have to do it in human cells, you can do it in animal cells, in plant cells, in any type of cell you want. So I want to conclude here. I don't want to take too much of your time, but if you would like to read more about the research in my lab, you can go to this website. And this slide shows some of the PhD students and master students that have done this work in my laboratory. And we collaborate with other scientists, both at Sabancı University in England, in Slovenia, as well as Boğaziçi University and other universities around Turkey. So I thank you, and I can answer questions if you have any.
Thank you, Professor Erman, for your speech. It was truly captivating. We're thankful that you shared your insights on genome engineering, a field that is revolutionizing biology, both in terms of scientific research and the advancements in technology it produces in various fields. We're certain that it was beneficial for our participants. With that being said, we will now proceed with the questions. You may send your questions to us through the Zoom chat or use the raise hand function to state your questions. Participants who are joining us from YouTube may write their questions to the chat there.
Maybe I spoke a little too long. Sorry about that.
Tunahan?
What are the future implications of genome editing that you expect?
You can change, once you change the genome, you can change the organism. Obviously, there are ethical implications. You can't really do this on humans. You may have followed the literature. There were some attempts early on in China to make genetically modified human babies. But I think we should understand that it's not, nature is not human-centric. There are many organisms that are used for agriculture, for the animal veterinary fields. And there are many things that you can do in nature, in laboratory that you cannot do in nature, but that allows you to understand this genotype-phenotype interaction. Of course, there are modifications. I didn't talk about this today, but there are derivative enzymes that can recognize not DNA, so CRISPR-Cas9 type enzymes that would recognize RNA and modify the RNA. Without really changing the genome, you can change the genome, the RNA sequence. And that's a different type of technology, which does not really create a genetically modified organism, but makes an organism that has a different phenotype. Again, like we saw with this COVID vaccine, you can modify genomes of viruses, and you can modify genomes of cells that are used to generate, using biotechnology, cells that are used to generate biological products much more efficiently. And it makes it much cheaper and much faster and much more popular that way.
I think we have another question by Batu.
Hello. So, what are the biggest challenges that stand in front of genome engineering right now, and how are they tackled?
Yeah, interesting. So, you may have heard of this off-target problem. So, when you design an enzyme that will bind to the DNA in a sequence-specific way, and it cuts the DNA. Of course, nothing is perfect in nature. So it may cut the DNA that you intended to cut, but it may also cut other places, which means you may generate mutations, unwanted mutations. These are called off-target mutations. So there are many techniques of trying to decrease the off-target mutations. And one of the techniques I showed, instead of making a double-stranded break with a single Cas9 enzyme, you make this nickase, so you cut twice, and you decrease the probability of cutting somewhere else, because it will only cut when the two enzymes are together. If one goes and binds, it will cut one strand, and the other one will cut another strand, and if they're not together, they don't make mutations. So that's one limitation, which is fairly easy to tackle. Of course, there are the ethical issues, but so far we've been using this for laboratory research, so not on humans. So we don't really make genetically modified organisms, but making genetically modified organisms, there's a big debate about whether it's beneficial or not. I think those are the main two limitations. What, in my experience, in my lab's experience, what we see is that as long this CRISPR-Cas9 enzyme is quite efficient. Like once you put the protein and the RNA into the cell, it will somehow find its way into the nucleus, and it will cut the DNA in every cell that gets transfected, or in every cell that contains the protein. The trick is to put this enzyme into the cell. How do you modify the cell to insert this protein RNA complex? That's another technology. We call this transfection, or you can use viruses to infect cells and transfer the guide RNA and the protein, the Cas9 protein, into the cell. So gene delivery is a totally different technology that has some limitations. But as long as you can put the enzyme into the cell, it will find its target and cut the genome, and the cell will, while trying to repair, will make the mistakes and make the mutations.
I think we have a question, Salim.
Hello, first of all, thank you for the presentation. I really enjoyed it. I have a question about the synthesis of the CRISPR molecule, I would say, because I think it's a combination of different things. So you said that the protein tails connect to different bases, and I mean it's a very hybrid, and it seems very complex, like the quaternary structure of it. How did they first synthesize it, in the lab?
Well, the CRISPR is actually quite similar. The CRISPR that we use in the lab is very similar to what exists in nature. So the Cas9 enzyme and the guide RNA. The guide RNA, we call it the guide RNA, but it's actually in nature it's two pieces of RNA. One of them that is the guide that binds to, that recognizes the DNA, and another one that sort of holds the Cas9 next to it. So there's this RNA-RNA interaction in the natural version, and then there's the RNA-protein interaction. And if that's all in place, then the RNA binds to the DNA and separates the two strands of the DNA, and the RNA binds to the DNA, and then the protein goes and cuts the DNA. So there were some modifications made so that it simplifies things. Instead of having two RNAs, you have a single RNA that's a little longer. I think you may be referring to the earlier technology, the TALEN technology. The TALEN technology uses these, in the natural version, has these repeats, 34 amino acids long and 18 repeats. So 18 times 34 is quite a long DNA binding domain, but each repeat recognizes one base. And which base it recognizes is determined by the two amino acids, the two amino acids in the middle of that repeat. And you can just change everything. So the 34 amino acids is always the constant. You just change the two amino acids in the middle, and then it will either bind an A, or it will bind a T, or it bind a C, or a G. So you have 4 repeat types according to which base you want to recognize. This is a very different way of binding. This is the helix that wraps around the DNA. The zinc finger nucleases that I talked about earlier uses a different thing. One zinc finger will recognize 4 bases. So it just shows how diverse nature is. The end result is the same. There's a protein that binds to DNA. Zinc finger nucleases solve this problem by having one zinc finger motif bind to 4 nucleotides. The tail proteins solve this problem by having one repeat bind to one base and then having multiple repeats. And the CRISPR-Cas9 or the Cas9 enzyme solved this by having an RNA connect with the DNA and having the protein sort of keep everything in place. So very different type of evolution, but it's kind of like convergent evolution, if you know what I mean. So using different structures to solve the same sort of biological problem.
Thank you very much.
Are there any other questions?
I actually have a question. Dr. Erman, thank you so much for your lovely, wonderful speech. Thank you so much for accepting our offer and spending time with us today on a Saturday, actually, during the weekend. And as a person who grew up with sci-fi movies back in the 90s, such as Jurassic Park and Gattaca, and those who haven't seen these movies, I highly recommend. I was wondering how close we are in terms of technology to the point where to these dystopic ideas of that they represent in the movie Gattaca as an example, where people are genetically modifying the embryo and maybe selecting the human beings according to their talents and abilities and the potential that they keep in their genes. How close we are to that future, or can we, as an example, protect endangered species through genetic modifications and engineering somehow?
Yeah, these are excellent questions. I mean, personally, I don't think we're anywhere close to making genetically modified humans. First of all, we don't really know where to start, with gene to, I mean, the connection. The problem is, in sci-fi movies, of course, one gene controls one phenotype, and in reality, it's rarely like that. So many phenotypes or many diseases, for example, are not controlled by a single gene, but multiple genes. So it's not like you change a gene, mutate a gene, and then you get like Godzilla or something. You have to, let's look at it the ethical way. If you want to mutate, you have a mutated cancer cell and you want to mutate it back to normal, back to wild type. Typically, there's more than one cause. There's environmental effects and there's genetic effects, but even the genetic effects, multiple genes control different phenotypes, individual phenotypes. So I don't think we're that close because we don't really understand the biology that well still. We don't understand how the brain works. We know quite well how neurons work and how they come together. There's a lot of neuroscience knowledge, but we can't really change and we can't really engineer yet neurons. We can change their genome to make them, give us a signal if we want. There's this field of optogenetics. So I don't know if you've heard about it. You make a transgenic mouse, and then this mouse has this protein. It expresses a protein in its neurons, and if you shine light, then the neuron starts firing, and then the mouse starts to go to the left or go to the right or something. So you can do simple things like that, but and as I said, there were these early reports that there were scientists in China that were modifying. Oh, of course, modifying human babies using Cas9, and I heard that it was the gene that makes you resistant to HIV infection. So they were supposedly making HIV-resistant Chinese babies. I don't know how true this is, but I mean, there's a whole knowledge about HIV and how it infects cells. And there's a protein called CCR5 that if you have a mutant version of the CCR5, you're less likely to be infected by HIV. So I think it's in the future yet, maybe 50 years or 100 years, something like that. But of course, science doesn't really work this way. There's this great book called the Structure of Scientific Revolutions by I forget the author's name. Does anyone remember? I don't know. But science works by, or scientific revolutions happen by not by slow evolution, but as we saw with the CRISPR-Cas9 field, by breakthroughs. So, you go for many long periods of time where you try to research and you try to find, collect information, and then there's this breakthrough. There's one scientist, one laboratory comes up with this technique, and then everybody jumps on and tries to use that model and make that model better. So we can't really see. I mean, 10 years ago, we couldn't envision a world where there was some guy talking about genome engineering. It didn't exist. There were people who were slowly working on zinc finger nucleases and talons, but nobody knew that in 2 years' time, every lab in the world, every molecular biology lab was going to use this enzyme called CRISPR-Cas9. So there's still a lot to be discovered, and because of these breakthroughs, something may happen next year that totally invalidates everything that I told you so far, and makes it much more easy to do genome engineering, and then the speed will be much more rapid. So not all science fiction stays in the movies. I think many science fiction that we grew up with is reality. Like these iPhones are, we couldn't really imagine that way science fiction did, but in some ways, science fiction gives you the imagination to create, to invent new technologies. So where art and science come together, maybe. So that's a long way of saying I don't really have a good answer to your question, unfortunately.
Thank you very much. That was all the time we had for questions. Thank you for your speech. We are sure that it will inspire many young researchers in the audience, especially those that wish to pursue a career in biological research. It was a very informative presentation.
Thank you very much. It was great to be here.
With that being said, Professor Arman, in order to express our gratitude for your speech and help pin our Turkish cause, we will plant 35 trees via the Tema Foundation on your behalf. Thank you again.
Thank you. Thank you very much.
We will now proceed with the second student presentation. Aybike Yeşim Eski Bozkurt will now present their research titled "To What Extent Soil Type Affects CO2 Concentration, Temperature, and pH in Mesocosm." We once again ask that our participants write down their questions. There will be a question and answer session at the end. Aybike, you now have the floor.
Hello, everyone. My name is Aybike Yeşim Eski Bozkurt, and I'm coming from İstek Acıbadem High School. Today, I will be presenting my study, Effects of Soil Type on Mesocosm, Temperature, pH, and Carbon Dioxide. Background: global climate change and higher demands of increasing population make the limited agricultural land even more valuable. Finding ways for soil and this ecosystem sustainability is essential. Monitoring variables in an ecosystem will tell us the point that we have to change.
Sorry to interrupt, Aybike. You can actually screen share the presentation.
I'm sorry.
We gave you the allowance. No problem.
Okay. Each soil type within its own ingredients has an effect on the ecosystem sustainability. Mesocosm. Let's talk about what is mesocosm. One of the ways of studying soil and ecosystem is mesocosms. Mesocosms are smaller and easily controllable enclosures that display the physical and biological characteristics of an ecosystem. On the right, you can see a picture of what a mesocosms look like. The aim of this study was investigating effects of soil type on mesocosm characteristics. We looked at three main variables: carbon dioxide concentration in water and air, temperature in water and air, pH in water and water. We built two mesocosms with different soil types, one with regular potting soil, another one with succulent soil. We used three probes to measure our variables: pH probe in water, carbon dioxide sensor in air, temperature probe in water and air. You can see our experiment design in these pictures. Contents of the mesocosm. Everything was the same between the two mesocosms except from the soil type. We used Genta brand regular soil and succulent soil. In both mesocosms, we use gravel, moss, Ottoman grass, evening primroses, ball succulents, star succulents, tree lentil seeds, elodea, and cotton in equal amounts. Here are the contents of the soils. On the left, you can see the regular potting soil contents. On the right, you can see the succulent soil. Now let's look about the results. First, we look at the carbon dioxide levels in succulent soil versus regular potting soil. Carbon dioxide is higher in succulent soil than the regular soil. Between start and finish, there is a 2200 ppm difference in carbon dioxide in succulent soil versus 1700 in regular potting soil. Then we look at the air temperature. Air temperature tracks similarly in both the succulent soil and regular potting soil. However, the succulent soil was higher in temperature overall. Then we look at the water temperature. Water temperature also tracked similarly in both succulent and potting soil, but on average, succulent water temperature was higher. When we look at the pH, we saw the succulent soil had a lower pH than regular potting soil. Therefore, the succulent soil was more acidic compared to the regular potting soil. Statistical analysis. The Pearson correlation coefficient was used to measure the strength of linear association between two variables. Correlation of paired trials were conducted for succulent soil data and regular soil data. Value R equal 1 means a perfect positive correlation, and the value R equal -1 means perfect negative correlation. Statistical analysis confirmed our graphical observations that there was a significant positive differences in carbon dioxide, temperature, and pH between succulent and regular soil. When we look at the Pearson correlation values within the soil types, we see that carbon dioxide leads to a decrease in pH, increase in carbon dioxide leads to an increase in water temperature, and these results were seen in both succulent and regular soil. Results. Carbon dioxide concentration in succulent soil mesocosm has a significantly higher carbon dioxide concentration compared to the regular soil mesocosm. Succulent soil mesocosm had a significantly lower pH compared to the regular soil mesocosm. These results were significantly P less than 0.05. Now let's talk about why the carbon dioxide is higher in succulent soil. This finding suggested that decomposition in the succulent soil is higher and contributes to more high carbon dioxide concentration. Air and water temperature was higher in succulent soil. This can be due to increased level of carbon dioxide contributing to a greenhouse effect. Water pH. Succulent soil mesocolumns has lower pH than regular soil mesocolumn. This is likely due to the elevated carbon dioxide levels in succulent soil, making the water more acidic. And limitations and future directions. The soils used in the experiment were obtained from the local market, and the exact mineral contents are not labeled precisely. This may affect the measurements that were taken. Further studies can address these limitations by measuring the naked soil productivity and decomposition without any other organisms. Finally, additional different soil types can be investigated for the same variables in future studies. Summary. In the summary, this study investigated the effect of two soil types on mesocolumns, air, and water quality. The results supported the alternate hypothesis that there is a significant differences in carbon dioxide concentration, temperature, and pH between mesocolumns of different soil types. These are my references, and thank you for your listening. If you have questions, ask me.
Thank you for your presentation. We can now move on to Q&A session. Participants may ask their questions verbally or send their questions as messages through the Zoom chat.
Mm-hmm.
Do we have any questions? We have a question in the Zoom chat. I can read it out loud so that everyone can hear. How can global climate change be solved by manipulating the soil type?
When we manipulating the soil type, it's affected by the other factors. So global climate change also, we can invest in different soil types for agriculture, and so that we have the more land. And yeah.
Aybike, thank you so much for your presentation. I was wondering, as a chemistry teacher, what is the biggest challenge you have faced with when conducting your research, when doing the experimentation? What do you think was the hardest part, or was it the research you made, or was it the part where you have collected the data? What would you suggest to young scientists who would like to maybe conduct a similar research?
Firstly, the hardest part is actually the tables, because making tables and getting factors of the mesocolumn is the harder part, and setting up the mesocolumn and was also hard, and finding data about the contents of soil. Luckily, I had amazing teachers to do this.
So as far as I understand, you're referring to the use of technology, maybe? The necessity of using technology when conducting research? And collecting data was the biggest challenge, maybe. Am I understanding it correct?
Yeah. Yes.
Okay, thank you so much, Aybike.
You're welcome.
We have another question in the chat. So if we could get a brief answer. the question is, how can we interpret the fact that succulent soil had more CO2? Could we say that regular soil is better for growing plants since the reason for a lower CO2 level might be a higher level of photosynthesis?
That's a good question. Each plant has different needs. So so depending on the needs, we will need different soil types.
Okay, thank you very much. At this point, we will be having a break since we don't have any more time. Thank you for your answers. We will now proceed with a recess until 6 p.m. Turkey time. After the break, we will listen to Batı Yalçın from Robert College in Istanbul. Please use this time to prepare any materials you will need during the presentation. Participants, you may turn off your microphones and cameras, but please don't leave the meeting.
So we'll reconvene in 5 minutes. 6 p.m. Turkish time, 11 a.m. Eastern time.
Hello, everyone. We will now be proceeding with the third student presentation. But before that, we kindly request that participants turn on their cameras. Batu Yalçın will now present his research about quantum computing in estimating ground state energies of atoms and molecules. We once again ask that our participants write down their questions for the Q&A session.
Hello, everyone. So I'm Batu Yalçın, and today I'll be talking about my research, which is titled Quantum Computing and Estimating Ground State Energies of Atoms and Molecules. So to give a brief overview of my presentation, first I'll be talking about the purpose of my research, and then I'll be moving on to the quantum mechanical many-body problem. What is it and how does it affect the classical computational chemistry, which I'll talk about later on. And then I'll be continuing with talking about quantum computation and how quantum computers can be utilized in the field of computational chemistry. And then I'll present my simulation and conclusions from that simulation. And then I'll end with talking about the future directions of quantum computers and their applications in chemistry. So let's start with the purpose of my research. Scientific breakthroughs in most areas that require chemistry, actually, but for example, drug discovery and energy efficiency require either challenging experiments or chemical simulations. For example, when I want to discover a new drug, I would want to simulate a protein which has lots of amino acids, and there are a lot of possible conformations for the protein, a lot of folding possibilities for the protein. And this is a hard task for a computer to simulate, even if it's possible. And most of the experiments are just impractical. However, quantum algorithms that use quantum computers, could make these simulations possible. I will get to what I mean by quantum algorithms later. In my work, I'm talking about the impact of quantum algorithms on the field of computational chemistry, and then the performance of quantum computational methods and comparing them. Let's take a look at what the quantum mechanical many-body problem is. This is a simple H2 hydrogen molecule. As you can see, there are proton-proton neutron nucleus-nucleus repulsions, electron-nucleus attractions, electron-electron repulsions, and then there are the kinetic energies of the nuclei and the electrons. And computing these individual forces are relatively an easy task because there are a little number of nuclei and electrons. Here you can see the Hamiltonian, the total energy, an operator that represents the total energy of a system, broken down into kinetic energy terms and potential energy terms, with electron-nucleus attractions, nucleus-nucleus repulsions, electron-electron repulsions, and the kinetic energies. However, when it comes to computing this for complex molecules like this, there are a lot of orbitals, there are a lot of electrons, there are a lot of nuclei. It isn't that easy. Let's see what Dirac had to say about this. The underlying physical laws necessary for the mathematical theory of the whole chemistry are thus completely known. He talks about quantum mechanics here, and the difficulty is only that the exact application of these laws leads to equations much too complicated to be soluble. Now let's look at what equations he's talking about. He's talking about the Schrödinger equation, which is essentially an equation which predicts the future behavior of a quantum mechanical system. There's another form of the Schrödinger equation, which is time independent, and it deals with stationary states, which are states whose observable quantities, for example, momentum, energy, velocity, are constant over time. And when it comes to the TISC, the time independent Schrödinger equation, the equation takes the form of an eigenvalue equation, in which we have the Hamiltonian, the total energy operator of the system, as the eigenoperator, the wave function, the function which describes the quantum mechanical system, as the eigenvector, and most importantly, the energy as the eigenvalue. We want to solve this equation and extract the energy, and especially the ground state energy, the lowest energy that the system could be in order to understand how that system would react with these different systems, for example. So how do we tackle this? There are classical computational algorithms for this. And I'll be talking about only the molecular orbital theory algorithms, not the density functional theory algorithms, for the sake of brevity. An essential algorithm is the Hartree-Fock algorithm. It tries to, all of these algorithms try to find an approximate wave function which describes the system accurately. And in real life, the exact wave function is the superposition, the combination of different electron configurations, for example, for a molecule. Hartree-Fock method tries to find the single best electron configuration that describes the system, and this makes it result in poor approximations. However, this does not mean that it is unusable, because the Hartree-Fock wave function is used as a starting wave function for more complex methods. Then there's the configuration interaction method, which takes individual electron correlations into account too, so it's more comprehensive, and it uses several possible electronic configurations. For example, the full CI in theory could find the exact wave function. However, this is also intractable because of the system size, and thus CI methods with reduced bases are used. For example, CI singles and doubles with only single and double excitations of electrons. And then coupled cluster methods come. They use a different operator on a trial wave function, which may be the Hartree-Fock wave function. It means just an initial wave function to obtain more higher accuracy solutions. For example, an operator might be a single and double excitation operator, which again excites the electrons to get another electron configuration and combines these and superposes these into the wave function in a different way. These operators are also usually truncated, again, to, for example, CCSD, singles and doubles, and this method is widely used currently. But if we have these methods, why can't these suffice? The memory and the time complexity of methods on classical computers grow exponentially, which we consider as hard with the system size, whereas the same qualities grow polynomially, which we consider as relatively easy in quantum computers. But how? An n qubit. The qubit is the unit of information in a quantum system, whereas the unit of information is a bit on a classical system, can produce 2 to the n states. Because, as you know the Schrödinger's cat, how as how a cat can be both alive and sorry, as how a cat can be both alive and dead at the same time, a qubit can be at the state 0 or 1 at the same time, so that it allows us to simultaneously work on different states at the same time. However, on classical computers, a bit can only occupy one state, 0 or 1 at the same time. So due to the classical nature, we can't do this on classical computers. And Google has demonstrated the advantage of this and showed that a quantum computer only needed a few minutes to perform a task that would take a supercomputer at least 10,000 years, not minutes, years. So let's talk about quantum computational chemistry now. Well, Richard Feynman said, nature isn't classical, damn it. And if you want to make a simulation of nature, you'd better make it quantum mechanical. And by golly, it's a wonderful problem because it doesn't look so easy. Here he's talking about if you want to simulate chemistry, you need to leverage quantum mechanical properties. And this is the quote that laid the groundwork for quantum computation. Let's take a look at what quantum computers are again. It leverages a superposition property because the qubits can be at the state 0 and 1 at the same time and collapse to 1 at different possibilities to increase computational power. As of hardware, there are FTQCs which are worked on right now but haven't been realized yet, and they are able to tolerate errors via error correction mechanisms. An algorithm might be quantum phase estimation that can be used on FTQCs for chemical simulations. However, the computers that we now have are noisy intermediate scale quantum computers, and they don't have sophisticated error correction mechanisms. They're intermediate scale, and they have been realized. But we prefer hybrid algorithms such as variational quantum eigensolver on NISQ devices. Let's talk about the VQE. The VQE algorithm first obeys the variational principle. In chemistry, it means that however you optimize the energy, you optimize the parameters and get the energy and try to minimize it, it won't get lower than the ground state energy. So we can try to minimize this function without the fear of going below the ground state energy. And it's a hybrid quantum classical algorithm, which uses quantum algorithms to compute the wave function and classical methods to optimize the parameters and again prepare this wave function. In my research, I simulated a lithium hydride molecule, which on IBM's noisy quantum computing simulator. And I obtained these graphs according to the interatomic distance between hydrogen and lithium. There are two things in VQE: ansatz, which is basically the way that we modify the wave function in quantum computers to obtain a better wave function, and optimizers, which are classical, which just update the parameters as a feedback mechanism. As of ansatz, I use two: hardware efficient, which leverages quantum hardware, and UCCSD, which is just a quantum computerized version of the coupled cluster singles doubles operator. And as you can see with the Kobila optimizer, which is a linear approximation optimizer, which doesn't use the gradient, I had great results actually with high accuracy, especially in the UCCSD. However, a point of concern here is the scaling of this classical optimizer with the system size. By using SLSQP, which is an algorithm that makes use of the gradient, I haven't had enough accuracy. The reason is an important problem in generally VQE and quantum computing, which is called the barren plateaus, which means that the gradient optimizer can get stuck in a local minimum and not be able to go down to the global minimum. And the gradient vanishes at different points. The reason is that. And when I used the SPSA algorithm, which is simultaneous perturbation stochastic approximation algorithm, I again got high accuracy results. Basically, what this algorithm does is it chooses random parameters. It chooses parameters in a statistically random fashion to optimize, and thus it doesn't require much system size because it simultaneously optimizes randomly chosen parameters, and it's performing with high accuracy also doesn't leave room to concern of accuracy, and the nature of it doesn't leave room for the concern of scale. So let's finally, before future directions, sorry, I'm having a bit of a problem with the computer. Let's go to the conclusions. The quantum advantage is born from the fact that the system of qubits that can simultaneously operate with an exponential number of possible states by leveraging the property of superposition, while a system of bits basically can't, and they can occupy only one state. And the quantum advantage has been empirically demonstrated by Google with 53 qubits in 2019. FTQCs haven't been realized yet, leaving us with NISQ devices, and VQE has been found as a suitable algorithm to use on NISQ devices. There are some challenges to VQE, such as the scaling of the ansatzes, scaling of the parameters, and the barren plateaus in optimization. I want to finally talk about the future directions. As I said, the VQE and quantum computation has a lot of applications in chemistry, but two stand out to me the most. First, energy efficiency. While with chemically simulating batteries, their efficiency could be drastically increased. And the companies in the QC industry, such a quantum computing industry, such as IonQ, are partnering up with automotive industry giants such as Hyundai in order to increase the efficiency of their lithium compound batteries by simulating them. And in drug discovery, simulating proteins and their folding is hard for classical computers because the sample space, the number of possible conformations is exponential. And in NISQ devices, possible conformations can be mapped onto a Hamiltonian, mapped onto the qubits, and simultaneously modified to find the most stable one with the help of VQE. The quantum algorithms are promising in the field of biochemistry, medicine, energy efficiency, and much more. And these are some examples of their applications. Thank you very much. And I want to especially thank Ms. Özilhan, my chemistry teacher, for giving me the opportunity for this research and assisting me throughout it.
Thank you very much for your presentation. We will now proceed with the Q&A session. Participants may ask their questions verbally by raising their hands or send their questions as messages through the Zoom chat to us. Again, participants joining from YouTube may use the live chat.
İpek, may I start, please?
Yes, of course.
Batu, thank you so much for your presentation. It was very detailed. And honestly, it was like over my head, such a hard topic to understand. And I was wondering, how long did it take for you to make the whole research? How much time did you spend to comprehend and understand everything going on around computational chemistry. If you could like give a couple of suggestions to your friends, your peers who are currently in the Zoom and online on YouTube, it will be great. Like, what will be your guidance for them in general?
In terms of computational chemistry, there are two main resources that I've used. First, there are lecture notes of several universities that are really detailed and really easy to understand, which helped me to understand the foundations of of computational chemistry. And then, after understanding the foundations, I read several of the recent and old papers that sometimes introduce these algorithms and sometimes improve them. For example, in terms of quantum computational chemistry algorithms, I both used a review on VQE and the first appearance of VQE as a paper to see the historical evolution and to have a better and more comprehensive perspective. And I must say, to learn quantum computing, I actually took a course, but that same method can be used for quantum computing too, because it is slowly integrated into curriculums of universities, and there are lecture notes of quantum computing that explains everything really well and simple. And then over time, again, diving into papers and reading about, actually, it's a hot topic, so reading in scientific magazines how quantum computers work and what advance we can see. Again, this may be a valid learning path.
Thank you. Islam informed me that she also wanted to ask you a question, so you may ask your question.
Thank you, and thank you, Batu. This was a great presentation. I particularly like the quotes you used. I want to ask, could you elaborate more on how does the superposition property of the qubits help enhance simulations in chemistry, or as you said, energy?
Okay, so I may give an example of Bell states, for example. Bell states are essential states in terms of quantum computation. Let me explain what they are. So you have two qubits, right? And then these qubits can be put in a superposition in which it has equal chance of collapsing into 0 and 1 when measured. What do I mean by this? It's both in the state of 0 and 1. It is counterintuitive, but it is like that. And there are different interpretations of quantum mechanics and superposition in general, and you may read on that too. But let's say that it is in the both states at the same time. And then when you measure it, when you observe it, when you try to learn which state it's in, it collapses into 1. For example, it collapses into the 0 state, or it collapses into the 1 state. And you can put this into a superposition in which this possibility is equal. And then when you, for example, entangle it with another qubit, I just realized I'm introducing another terminology here, but when you intertwine their fate with another qubit, I don't know how to explain it, but when you make their actions depend on each other, I may say, entangle them, you have the possibility to modify a 2-qubit state by which has different possibilities. For example, a 0-0 state and a 1-1 state at the same time by modifying 1 qubit. And in this simple way, you can work on 2 states at once. In a bigger perspective, there is this Grover's search algorithm in which you try to find something. It's just a general computational search algorithm. For example, there are 8 options, and you try to find one according to some set of rules. In Grover's search, for example, you actually combine these states in a superposition. You modify this superposition, and the probability of the right one is amplified at some point. And when you measure it with a lot of times, due to the law of large numbers, it collapses into the right state. If you weren't able to do Grover's search, you wouldn't be able to modify all the possible states at the same time, simultaneously, and then find the right one with that.
Thank you.
So there's a question by Salim Sinan.
So hello again. My question is, you talked about Baron-Platos, and I was meaning to ask, the way you explained that it's seemed that it's rather more trivial than the things you've dealt with. So what is the problem with barren plateaus that makes it so hard to overpass?
So in general, in chemistry, for example, while finding ground state energies, let's draw just a random function. Oh no, I should draw it the other way. I'm trying to find this here. Let's say this is the ground state. However, when I use a classical optimizer to optimize this, because there's a lot of parameters, like if you think of a single variable function, yes, you even like a human can take the derivative and everything. But when there are a lot of parameters and the scaling is large, when we make the computer do this and use a gradient-based algorithm, which is one of the most suitable ways, because optimizing the function with a gradient is much more easier. Here, for example, there's a local minima because the gradient here is again 0, and the algorithm is tricked into thinking that it's actually found the minimum. And this, why this is hard to deal with is that we don't have that much control on where, like, we can't say, okay, this is a local minima, just skip this to the algorithm. And the algorithm just tricked into this. And this isn't just in chemistry. This problem is especially evident in quantum machine learning too, and the solution of escaping barren plateaus. Because as you know, in machine learning, deep learning, the cost function optimization while using the gradient basically means optimizing the error with the help of the gradient is a widely used, a ubiquitous application. If we get rid of this problem, the future of quantum machine learning would be brighter than ever. And the reason is, as I said, the function is more complex and multivariable function in which we can't interfere with the workings, and it's generally hard to manipulate consciously.
I see. Thank you.
So are there any other questions? It seems that there aren't, so thank you very much.
Thank you for your answers once again. We will now proceed with a recess until 25 past, after which we will yield the floor to our keynote speaker, Professor Naz Zeynep Ataygök. Participants, you may now turn off your microphones and cameras, but please don't leave the meeting. We will inform you once again when the break is over.
Hi, Professor Gök. We now have a recess for 2 minutes. In the meantime,
Okay.
you may prepare your presentation.
I'm prepared. Can you hear me?
Yes, we can hear you.
Okay, I'm going to use these. If you lose my voice, okay, tell me so I can take off my earplugs. Okay.
Okay, thank you.
Okay, I'm all ready. Whenever you say go ahead, I will share my screen. Okay.
Okay, we have a 2-minute short recess, and that's only for 2 minutes.
Oh, okay. Sorry, you said you have a 2-minute break.
Yes, until 25 past.
Okay. Do you want me to say anything in between, or shall I wait another 2 minutes and everybody
We
will
can
be back?
directly proceed after 25 past when everyone's back.
Okay, okay. Okay, have a break. I just read your 5 dakika ara verelim dedi. Do you want an introduction, or shall I do it? Shall I introduce myself?
We actually have an introduction in a few
You're going to
minutes.
do that.
Yes.
Okay. Do you choose
And you can
Robert
also
College?
continue. Yes.
Okay. Do you choose Robert College graduates especially, or just randomly? Anybody could be a talker.
Yeah. Anyone could become a keynote speaker, but like
I
I
mean,
don't
do
think
you
we
know
had
that
a
I'm?
particular. We knew that. Yes. We
You
knew
knew that. Okay. Okay. Okay. Okay. I'm just waiting. I'm quiet. Just tell me whatever you want to say. Okay. Just tell me to share my screen whenever you want. Introduce me and then do whatever, you know. Hi.
We can come back. We kindly request that participants turn on their cameras before we proceed with the keynote speech. We would like to introduce our keynote speaker.
Naz Zeynep Ataygök, a 1980 graduate of Robert College, graduate degree in chemistry from University of Kent, England, and started her academic career as an assistant professor in the Department of Chemistry, Faculty of Arts and Sciences at Boğaziçi University in 1987. She became associate professor in 1994 and a professor in 2000. She is still working in the same department as a professor of physical chemistry. Her research interests are in the field of surface chemistry, especially organized structures such as micro-nano-emulsions, vesicles and nanoparticles, and their application in transporting various chemicals encapsulated in these structures. Professor Ataygök also had various administrative positions at Boğaziçi University, including registrar, summer term coordinator, dean of student affairs, the director of School of Foreign Languages, director of International Affairs Office, corporate director of Erasmus Plus and Mevlana projects, vice rector of Boğaziçi University in charge of students. Besides, Professor Ataygök is currently the head of the Department of Chemistry. Professor Ataygök, you now have the floor.
Thank you. Thank you. I hope I don't lose you. You can hear me. Just warn me. And in case something happens, please give me a phone call, okay? Because I'm not in my own house at the moment, so I might lose the connection. So I might have to reconnect something. You know, if something happens, just let me know. Okay. Thank you. Thank you for the introduction. Okay, that's all I would have said. I think I sent you that introduction, right? I'd like to share my screen and tell you and take you to my wonderful and extraordinary world of surface active agents, which I have been working since the 80s, sort of mid 80s, I should say. That shows my age and everything. Okay, let me share my screen. I hope I can find it. Okay, here it is. I'm going to go to the presentation. I work with surface active agents, also called surfactants, surfact, A-N-T-S. It's a short version, I know. But we use surfactants in more or less, you will see that we use surfactants more or less in everyday life. And I will take you to that world and how we have been working with them in the laboratory, but also in the everyday life products. I'll just remind you, okay, I don't know, I'm pretty sure you're familiar with all these things, but I just want to make a short introduction on polar and non-polar molecules. I'm looking at my screen and you can see that, by the way, when I was preparing these, I took a lot of pictures from Googling the web. So I put the references of these pictures, not the writings, but the pictures. If anybody's interested, you can go and check them out. But if you have a polar molecule, that polar molecule meaning like ionic compounds, they carry charges generally. They like to dissolve in polar solvents, such as water. Water is a polar solvent. It does have positive and negative charges, oxygen negative, hydrogen positive, etc. Non-polar molecules, such as the hydrocarbons, do not dissolve in water, okay? They dissolve in non-polar solvents, such as in benzene, in hexane, or, you know, all sorts of apolar or non-polar substances dissolve in apolar, non-polar solvents. Now, my molecules, the surface active agents, are both polar and non-polar. So what you see here, this is a very rough representation, but the molecule has got a polar head and a non-polar tail. This is a very, as I said, this is a very rough representation, but we usually call that polar head likes to go into water. That's why I put it as a hydrophilic head group. So hydro for water, philic to love, as you know. So that head, the polar part, likes to go into the water or into any polar solvent. This part, hydrocarbon, for example, containing 8 carbons and so many hydrogens, etc., that doesn't like going into water. It doesn't, you know, it doesn't like it at all. So it goes into apolar substances. Okay, what happens to this molecule if it's at an interface? Interface, obviously, in between two phases. Surface is an interface. Surface, as usually when one of the interfaces, one of the sides, one of the phases is air, we call it a surface. But an oil-water surface is called an oil-water interface. These molecules, okay, some of them do go into water, some of them do go into oil, but they love to orient themselves with their tails going to the apolar oil and their heads going into the polar water. So you see these wiggly surfactants at the interface. They like being in these positions, okay? Next picture shows that's another one from the Google. Okay, this is what happens with air. Again, the heads like to be in water, the tails like to get away from the water. So air is a good option. So they go into air. And then if in water, if you reach a certain concentration, what we call a critical micelle concentration, a certain critical concentration, they form rather strange structures where they somehow minimize this tail contact with the polar water. What I'm going to show you, look at my hands, if you can see me, my fingers, okay, they go into a spherical structure like that. I put on this ring, especially for this lecture, because this ring is pretty big, etc. But imagine that's the head of a surfactant, okay, sticking out into the water. And these, my fingers are the tails. They go into that structure and they close up. So water cannot go into the core, okay? This is a micelle. So I'm going to talk, obviously you must have seen them, you must have heard of them, but that's the idea, okay? They minimize, because they minimize the energy of bringing these molecules in contact with water and themselves, okay, by doing this. So you see these surfactants. Surfactants are the active ingredients of all the detergents we use. You know, I took some pictures. This is from my kitchen, actually. Pril, Omo, Ariel, Yumosh. There is a difference between the Ariel, the washing powder, for example, and Yumosh, which is a softening ingredient. I'll say a few things about that in a second. But these are these surfactants are active ingredients of all the stuff that you use in everyday life. Okay, I'll give you some more examples. How do these detergents clean the surface? How do these surface active agents clean the surfaces or interfaces, let's say? This is dirt at an interface. Imagine you have your plate with the dirty food. These molecules in Pril or whatever you are using to wash them off, okay, and it works for your shirts as well. The same mechanism works. They come and stick onto the dirt. The dirt is going to be rather hydrophobic, actually. So the tails stick up to that, and they lift up this dirt, just like the micelles they form a structure just like the micelles you've just seen, and they take it into their core with the heads in the water, and then they just carry it off. And the interface is actually covered up with these funny molecules, these extraordinary molecules. So that stops the dirt from getting reabsorbed at the interface, okay? So what do you do with this? You rinse it off, don't you? You don't just put some surfactant, some detergent onto your shirt, onto your plate. You do it, you wash it, and then you wash it off, okay? So that's the idea. So the basic, very simple structure of the dirt removal is this. That's the mechanism. Okay, there are other things involved, obviously, but I'm trying to sort of simplify and show you what is happening. Obviously, this is not the only thing they do, okay? You also see a lot of these surfactants in a lot of other products. Walk into a pharmacist, hair shampoos, cleaning waters. I especially chose these, some of them, micellar makeup remover. About 20 years ago, you would never see this micellar stuff in the pharmacy, okay? Kusursuz makyaj temizlemesi, whatever it is, okay? But it seems that somehow myself seems to have got into all these, not in all of this. And there is also some products you will see as you walk into the, I'm sure, just pay attention when you walk into the pharmacy, you will see some liposome containing products, cosmetic products, cleansing products, etc. So all these things all of a sudden have, okay, these sound fancy, but they're not any different from the products we used to use. Well, they've been improved, obviously, but they're not really that much different from the products that we have been using for 30 years, 40 years, 50 years, maybe. Okay, so we see these products, these surfactants, the ingredients of the detergents, active ingredients, sorry, can be classified as, one of them is ionic surfactants. Look at this molecule. This is old hydrocarbon chain, long chain. There is a nitrogen here. There's a positive charge on it. There is a counter ion, bromide, okay? But this is a positively charged head. So we call it a cationic surfactant. Cetyl-trimethyl ammonium bromide, CTAB. I've worked with this in the lab quite a lot. Long chain, negatively charged. Look at the negative charge on the oxygen. Okay, negatively charged head. This is an anionic surfactant, but each surfactant has got its counter ion to compensate for the neutrality in the solution. Sodium dodecyl sulfate, which means instead of oxide, you have dodecyl part in here, is more or less in all the, just look at the back of your shampoos, you will see that it's more or less in all the shampoo products that we use, okay? So when the head is charged, we call them ionic surfactants. Well, it's also possible to have non-ionic surfactants. Just one thing before I carry on. Remember, Yumosh, I said, is a softening product, okay? Yumosh is cationic. And Ariel contains anionic and maybe zwitterionic. It's a mix, it could be a mixture, but these cationic surfactants are especially used for softening products, okay? Hair shampoo is different, but hair softeners, I don't remember any names, brand names at the moment, they contain cationic surfactants, okay? So they do have different actions in the process. Non-ionics, okay, these are surfactants, they go onto the interfaces, but this is an example. They do have certain, they do interact with water, obviously, but it doesn't have any charge on it. Non-ionics, nonoxynol 9 and 9, zwitterionic. Okay, nitrogen has got a positive charge and the oxygen has got a negative charge, so it's doubly charged, but it's not the same type of charge. Opposite charges are present within this surface active agent. So we call it zwitterionic surfactants, which are very commonly used in baby shampoos because they stop ice thinking. You know, the babies are very prone to, they don't like being washed because when the shampoo gets into their eyes, etc. So it has to be something like that. So now you have an idea about these. This is a surfactant, a synthetic surfactant that I have used. Most of these that I have shown are synthetic surfactants. They're prepared in laboratories. But there are also obviously surface active agents that are naturally occurring. This is a doubly chained anionic surfactant. It's called AOT. It has nothing to do with, it's called aerosol OT, but it's not an aerosol. Can you see this double chain? A long chain and another long chain and the head group here, okay? So there's a reason why I'm showing you the structure of this doubly chained surfactant, because what these surfactants, what these molecules, extraordinary molecules do depend on their packing ratio, okay? P, I would call it. In most of the books, you will find it as P. It's a simple equation, V over A naught times L. Now, A naught is the area of this hydrophilic head group. Volume is V is the volume of the tail group, okay? But it's the chain because it's the hydrophobic part. And L is the optimum length. As you can see, I have drawn this with a wiggly tail because it's not very straight. You don't pull the chain. The carbon, hydrogen, the structure is not all very straight, okay? So it's the optimal length that we're talking talking about. And the volume is not just geometrical volume, it's the total volume it occupies. Now, I'll show you how that works. Look at this sodium dodecyl sulfate, very similar to the sodium octyl sulfate that I have shown you. This has a tail of 1.7 nanometer, more or less. The volume of it is about 350 cubic angstrom. The head group, area 58 square angstrom, okay? That sort of thing. That means it has a relatively large head and pretty low volume tails. So you see, it sort of forms a conical shape. Whereas if you go back to my AOT, the surfactant that I have worked with pretty much, okay, it has a double chain, just as I have shown you in the previous slide. The head group is not that much. I'm sorry, these are actually the same thing, 51 square angstrom or 58.58 square nanometer, same sort of thing. They're more or less the same size, not too different, okay, with respect to the head group, but the volume of that the tail occupies is much larger. Look at the different shapes that we can draw, you know, when we try to draw something schematically. Look at the differences. Well, okay, all that packing ratio, the volume to head group, etc. ratio, determines how these molecules behave at interface. Okay, I'm going to pull you aside. When the ratio is less than, let's say, 1/3, which means, or much larger. Okay, this means volume is small, the denominator is large. Okay, the head is large, length is large, but the volume is relatively small, just like in this case. Okay, the top case. They can, they can fit into, sorry, I missed that. They can fit into these micellar structures, what we call normal micelles, with my fingers. They can fit into that structure. But as the volume gets larger and larger, they open up because they have difficulty in fitting into confined structures like the normal micellar spherical structure. So they get bigger and they open up, and somehow they go into these open bilayer structures, bilayer, 2 layers. Look at the structure. Do you see that the volume is much larger? So it's all blackened to show you the volume occupied. Vesicles, flexible bilayers are in when the ratio is in this order, more or less. Planar bilayers are like that. Bilayer, outside is water, this side is water, but inside is the hydrophobic tail. So pretty much hydrophilicity outside, hydrophobicity inside. Plain bilayer. I will come back to bilayers in a second. And once they get bigger, like the AOT case, when the tail gets much bigger, or it occupies a larger, volume, then they no longer fit into the inside of the micelle. Instead, they reverse and they form reverse micellar structures, which means you now have the heads inside the structure, heads that, like water, go into the core, and the tails go out. Obviously, you can imagine this takes place in an oily environment. This takes place in a watery environment, aqueous environment, because of this different structures of the tails and the ratio of the head to head and tail and area of the head group, etc. Okay, that ratio we're talking about. So they're able to form very different structures, extraordinary structures, depending on their own chemical structure. Okay, big tail, small tail, etc. So let's go to the next one. This is again the same thing. I'm going to show you. This is the sort of representation you will find. This is a very famous picture. You will see, look at the head groups looking out, And this sort of, the radius is more or less 1 nanometer for a normal micelle. Okay, we're talking about nano dispersions. The interface, the outside, and outside is water, inside is pretty much hydrophobic. And in a reverse micellar structure, it's the other way around. It's the core contains some water, and the outside is apolar, and we have an interface here, okay, where the heads and the tails come together, etc. Okay, so interesting. Why am I, okay, I put this again to show you that this is more or less in the nanometer, well, it is in the nanometer range, and we talk about them as micelles or nano droplets, etc. Okay, what's the idea? The idea behind all sorts of research that we have been carrying out. I mean, obviously, I start off with small micelles, reverse micelle structures. We're onto more complicated structures, but the idea behind it is still the same. Look at this. If you put a hydrophobic dye, something that doesn't dissolve in water, something that doesn't like water, you put it into water, it sits right in the bottom. Take a little bit of, I don't know, take something that is hydrophobic, a little bit of dye, put it into water, it goes and sits in the bottom. You start adding surfactants to this. And somehow when a certain concentration is reached, these surfactants come together, as I have shown you before, you know, they form micellar or other structures. Now you have seen there are other structures. Because the inside, this part, is hydrophobic, this hydrophobic dye goes and sits inside there. So that means all of a sudden your transparent solution, the aqueous solution, okay, I've colored them red, but overall, when you look at the solution, you will see that it's in red color because all these droplets that are dispersed in water contain these dyes inside, and now they can carry this dye through a medium in which it is not soluble, which is very important. That's the idea behind more or less all the drug delivery systems, etc., that has been developed since the 70s, 80s. They carry, these are, obviously, I'm just showing you the very simple micellar structure, but all these structures, they encapsulate something that is not soluble in the outside, okay, in the continuous phase, as we call it. These droplets are the dispersed phases, okay, these structures. So the dispersed phase, which is inside the continuous phase, carry the stuff that is not possible to carry from one phase to another otherwise, okay? So they act as carriers, encapsulators. You will hear this terminology quite a lot: encapsulation of drugs, encapsulation of this, that. So that's the idea, okay? So how can we see this? Absolutely. The moment they're all absorbed, they're all encapsulated within the cores of these strange structures, you start seeing an absorbance. At that certain concentration, these structures form, they absorb whatever. I hope I have still time, okay? Okay, this is the idea. Water in oil emulsions or microemulsions, what I've been saying, normal micelle. You start adding oil to a normal micelle, the oil goes into the core. And you add more and more, it goes further into the core, okay? And this structure gets enlarged, okay? We call these micelles in water. This is an oil in water microemulsion. When I started working, it was called microemulsions. Now it's called nanoemulsions because the size is in nanometers. Okay, so oil in water, oil is in water. Microemulsions, when they get larger, oil in water emulsions. Just like here, this is a nanoemulsion, maybe a microemulsion, bigger. And when it becomes an emulsion, it means a dispersed oil phase or the dispersed droplets are so big, it's no longer transparent and it becomes milky. That's when you know you have very large droplets, and that's the emulsion, okay? And obviously, milk is an emulsion. Examples to oil in water emulsions: milk, mayonnaise, ice cream, salad dressing, chocolate, some of them. Example is emulsion of chocolate. Chocolate is actually in the, it should be in the other page, but anyway, it's stuck here somehow. I made a mistake, I think. But anyway, these are all oil in water emulsions. Yeah. Water in oil, the other way around, is you have reverse micellar structures, water in oil microemulsions, water in oil emulsions, okay? So again, the same thing. Sorry, chocolate is here, actually. I should get rid of the other part. Water gets dispersed. Butter, margarine, these are all emulsions. There is water in them. It's not just oily stuff, but it's a mixture of the two with some surface active material that binds them together. What we do at the moment, what we have been doing, I'm just going to sort of memorize. Am I doing okay? You're following me? Any problems? Okay, and stop me and ask me questions if you want, but I want to sort of, I'm trying to cover a lot of things, okay, but I hope it's okay. Liposomes or vesicles, this is what we've been working with. Do you remember I showed you the structures? In the beginning, they formed this conical ones, went into normal micelles, and then they enlarged, went up, and then they were getting larger and bilayers were being formed. And I said vesicles. Well, okay, these are the two words that are used interchangeably. Okay, I sometimes call them vesicles, sometimes liposomes. Actually, there is a slight difference, I think, but nobody really, I mean, if you come across any of them, they're more or less the same thing. Okay, so what we have in these vesicles, or the liposomes that I'm going to be talking about, can you see these extraordinary structures? As you can see, it's in water because the head groups are sticking outside. Tails, okay, and look, there's another tail looking up, another head group. So the core is aqueous. Interface, bilayer, two layers looking at one another, and another head group area, region, okay? So obviously this is aqueous, but this is non-aqueous. It's not likely to contain any water. Maybe there are some water molecules, but that's it. So this is what we have, a bilayer structure, okay? This is quite nice. This is the same thing again. Many naturally occurring and synthetic surfactants and phospholipids. That's another thing. That's the natural stuff that cannot aggregate to form micelles when dispersed in water, will spontaneously form closed layer structures known as vesicles or liposomes. That sort of structure. They cannot. They're too big to form micelles, but you know they don't go into flat bilayers, but they can form round spherical structures like these. Vesicle is a hollow sphere created from a bilayer surfactant structure with water media both inside and outside. Aqueous core is separated from the bulk aqueous, the outside medium, by a hydrophobic membrane, that part, okay? So as you can see, again, these can be used in encapsulating a lot of stuff. Vesicles, you might come across these. Actually, when vesicles form, they're like onions, you know, layer by layer by layer, you strip off a layer of onion and then you come to it until you come to the core of the onion. Okay, these are multilamellar, multi-layer vesicles. Can you see each one? Inside core is aqueous, and then bilayer. There's an aqueous line here, and then another bilayer, and then an aqueous region here, and then the bilayer, etc. It goes on like that. LUV and SUV, this is what we work with SUV. Small unilamellar vesicles, large unilamellar vesicles. This is the one we work with. These are what we call liposomes, actually. Small and unilamellar. So there's only one aqueous core, one outside, and one bilayer. Okay, so you can start off with this, and then you can get rid of all these layers like you are, peeling an onion, and then you end up with the inside, with the single unilamellar vesicles, okay? Phospholipids, these are lipids, okay? And okay, these are naturally occurring ones generally, but you can have synthetic ones obtained from the labs, obviously. But can you see they are also surface active, all these lipids, because they have head groups. In fact, this is a Zwitter ionic one, isn't it? And then long doubly chained tails. This is another representation, or that one, the MPG or the MPC. We usually use them together in some of they combine together the ratio. I mean, one bilayer doesn't have to consist of one type of phospholipid. You can have more than one type of phospholipid in forming these vesicles, and this is a combination that we have used in one of our researches. Okay. Now I'll show you a biological diagram again. From that reference, I thought that was one of the best pictures that I found. This is a cell membrane. Now, do you recognize the surface active agents and the hydrophobic tails and the hydrophilic head groups? This is what your skin is made up of, your whole body, okay? So this is the cell membrane structure. There are proteins, integral proteins, lobule proteins, carbohydrates, cholesterol in between. Cholesterol actually ensures the stability of the lipid membrane, okay? So that's the idea. How am I doing with time? Okay, I'm going to go quickly. But can you now recognize what I've been trying to show you with respect to the cell membrane? Okay, so skin compatibility. Why are we using these vesicles? Because they're compatible with the skin. When you put something inside these vesicles, which we are trying to do, which we have been doing, and a lot of groups are working with encapsulating, that's the term, encapsulating different things. People encapsulate drugs, cancer research is heavily involved in this, drugs, DNA, all sorts of things you put inside these vesicles, and they can, not easily, but they can penetrate through skin. They can release their cargo, whatever they are carrying, into the skin or below the skin, depending on where they want to send it. Or you see, it is compatible with the body when you find the right formulation, because not all of them would be the right formulation. This is back to these original. Remember, I said liposomes. You will see in cosmetic stuff, because we've been working on something like this. People use these liposomes, vesicles, but liposomes at the same time, same thing. They are, because of their skin structure, skin compatibility, it makes them ideal carriers for active drug or cosmetic ingredients into the skin. Okay, that's the idea. Again, another phospholipid and lipid vesicles, just to show you another picture. Okay. We've done very initial work we've done with the vesicles these were synthetic vesicles. We encapsulated cytochrome C, and then we broke these down, made little micelles, but we were able to release these cytochrome C that was encapsulated. That was a few years ago. We saw them being released, just like the drug being released into the organ or the region that you want to release them into target delivery. Okay, we don't do that at the moment, but target delivery and drug delivery is a very important part of the research in this area. Okay, what we are doing at this moment, present work, is transdermal protein delivery. So instead of drugs, we try to deliver protein. Studies for atopic dermatitis treatment. This is eczema, by the way. Okay, so we're trying to find the cure for eczema. Okay, I hope we do. I just put a couple of pictures just to show you. You see, this is the cell membrane. So whatever is being carried is going to be released through there. So that's the idea. Can you see the pictures that how whatever the cargo is being released through the skin, these pachymides, the inner aqueous core, etc. Exactly the same thing. I'm not going to read through these, but I want you to see how it's been done, and that's the idea. Okay, but we are not actually, okay, we've done these with lipids, but we've actually gone a bit further. At the moment, we're using not lipids, liposomes, using lipids, I meant, hydrophilic tail, etc. This is the liposome, or the vesicle that I've been talking about. It has a cargo, but now we don't use niasomes. That's another thing. What people have realized that these need, some of these liposomes are not flexible enough. They do not match with the skin as well as you would like them to be. As I said, you have to find the right formulation. There is something called ethosomes, which we've worked with. That is, there is ethanoic solution of the drug. It's not water now, it is surface active agent, ethanol, and phospholipids, okay, ethosomes. There is also something called transfersomes, which we are working with at the moment. It's another formulation, it's another modification of these liposomes. This time, there is not just ethosome, not just ethanol, but also edge activators or surface enhancers or whatever. You just start adding things to the interface to make life easier, to make them more compatible with the skin structure, etc. 1901. I think that was my last. So, we're working on this sort of thing. And thank you for your attention. And then I can stop. Okay. Was that okay?
Yeah, thank you for your delightful speech. Learning about the chemical interactions that underline the properties of surfactants was thought-provoking. We now have the Q&A session for you.
Okay, yeah.
May ask their questions verbally by raising their hands, or they can write their questions through messages through the Zoom chat. If they're watching from YouTube, they may use the chat function also.
Certainly, I'm used to that. A lot of people watch my classes from the library. They cannot speak. They write the questions in the. But I think Aras is raising his hand. Do I start by asking? I saw yours first. Aras, would you like to start off?
Okay, of course. So you have mentioned that azevitol-ionic surfactants are mostly used in baby shampoos because they don't hurt their eyes. Mm-hmm. So this information actually inspired me to ask, what are the main factors affecting the health friendliness of surfactants, actually?
It's a little bit difficult to say. This is a generalization, obviously. Not all azevitol-ionic surfactants can be used in baby shampoos. Non-ionics, because ionic ones, look, surfactants, as you have seen. Now, we're trying to interfere with the cell membrane, okay? But surfactants actually are dangerous to the skin, as you can see. You're not supposed to drink shampoos because they can damage the cell structure. We're trying to damage the cell structure to release something inside. Do you see what I mean? But the idea is not just that. You have to find the right formulation. I know that azevitol-ionic ones, for some reason, maybe it's because of their neutrality. I'm not, to be perfectly honest, I'm not an expert on that, but I know they are being used. But you see, surfactants are at the same time quite dangerous molecules because they can interfere with the skin. From time to time, so I cannot really answer your question, what makes it a better one for that. Obviously, it is after a long term of research that they have realized that this is a less stinking, but not all azevitol-ionics would do that. Certain ones. Obviously, they would be they probably are hidden somewhere. We don't know the exact names of those molecules. But obviously, not all the molecules are that safe. From time to time, you will get messages saying, don't, SDS, sodium lauryl sulfate is SDS, sodium dodecyl sulfate, it's the same thing. If you drink it, you'll die, blah, blah, blah. Yeah, but don't drink it because it's supposed to be used topically. Do you see what I mean? Probably another thing could be these deuterions might be washed up quickly. That's another thing. They don't stick to the eye. Maybe that's another. See, because some of them stick to the surface more than the others. So when you're trying to do something, create or form a new formulation, you probably have to look at that. Do you want it to stick or do you want it to move away quickly? That sort of thing. Do you see what I mean? So probably it has something to do with the light charge it carries, charges it carries, etc. Okay. So basically, they're not that safe molecules, basically because they do interfere with the cell membrane. But if you use them topically, like shampoo, no problem. Don't drink it. Was that okay?
Yes, thank you.
Okay. I had another hand, Tunahan, but I think Benangula, were you before? I cannot hear you.
Yes, I was. Thank you for your presentation, lovely presentation. I would like to ask you a question about interdisciplinary research, your idea about that. I know that you're in the search of chemistry, but do you study only with the chemists or
absolutely not? Absolutely not.
I'm a biology teacher.
Absolutely, you're in the right spot. Yeah.
I know one of my students, previous students, was studying on exosomes, which is also related to your area. And you answered most of my questions, but I know that exosomes, and I'm sure that the liposomes that you have talked about that, important for cell signaling. And do you think they will help us in cell differentiation in stem cell research also?
Cross our fingers. I hope so. I certainly hope so. But you're absolutely right. This is an interdisciplinary field, totally. And we do work with, I have done some research with we're still trying to publish it, but we haven't. We have encapsulated DNA fragments and they lived inside them. They were still active, etc. I have worked with the biologists, molecular biologists at the university, but a lot of people work together. You have to work together with other departments in these fields, okay? Because you have, absolutely, you're right, because first of all, these are great carriers. They carry any sort of cargo. They carry aqueous or hydrophilic cargo, hydrophobic cargo, or anything that doesn't like oil or water, but like to be at the interface. So they are universal carriers of stuff, okay? They can carry, of course, you have to find the right formulation, right ratios, right size, right surface active material. And as you can see, we have started working with synthetic material, but the real research is really with obviously in the lab, you have to use a lot of synthetic material to come to a conclusion, but yeah, you have to, yes, people use real life or natural surface active agents. And yes, they will be quite helpful with the type of research that you're talking about. This is not a chemistry field at all on its own. This is a totally interdisciplinary field. You have to have physicists with you, you have to have theoretical people with you, you have to have molecular biologists with you, any sort of scientist.
Thank you.
That would be the right word, I think. I think Tunahan Tarakçı had a question.
Yes, my question is, are there environmental methods to produce surfactants or surfactants that are environmental friendly?
Okay.
Do we have enough research made to investigate the long-term effects in
Oh.
nature?
Yeah, I didn't go into that actually. I actually teach colloidal science, and this is part of the colloidal science. Colloid, as you know, is one phase dispersed in another. And when the people start to work, now we do use biodegradable surfactants. People didn't know about them when they first started using this. By the way, you all use soap, don't you? Soap has been around for 3,000 years or so. So there's soap is a detergent. But when we say detergent, everybody understands something synthetic. What I'm saying is they all contain surface active material. So everything synthetic or non-synthetic, they're all surface active, they all contain surface active material. When, for example, I believe it was in the 50s in the United States, people realized that from the rivers and sewages, etc., came up froth, bubbles. Something was happening, something was going drastically wrong, and they didn't know what was happening. And then they realized that all the surface active material, all the detergent, I'm going to say detergents because surfactants are just the ingredients, the active ingredients of those, they were being, washed into the sewage, and then they were being drifted to the rivers, and everything was being polluted. So that caused a big problem. And then research started from the 50s onwards to make sure that we should have a biodegradable surface active agents in the stuff that we use. I'm just hoping that we are all using that. But the clandestine type laboratories, the Van Alta as we call them, you never know what they use, okay? And how that might damage the surface, how that might damage the environment. You're absolutely right. It's, as I said, it is an extraordinary molecule as a molecule. Surfactant is an extraordinary molecule, but it has its positive and highly negative parts to it, okay? What we are trying to do is use natural surfactants, natural surface active agents, but the synthetic ones especially have to be made biodegradable. So what I mean by biodegradation, I don't mean the molecule disintegrates. The molecule loses its surface activity, okay? Which means it doesn't go into the interfaces. It can be dissolved in water or it can be dissolved in oil, whatever part you want to look at it. Do you see what I mean? It has to be degradable. In other words, it has to lose its surface activity, okay? You're right, it's a dangerous field and you have to be very careful, and you have to, as a concerned environmentalist, I'm sure you all are, you should look into these things and make sure that it's not being harmful in any way to the environment. But am I ever going to stop using detergents in my washing machine? Probably not yet, okay, because I want to wash things off. Something psychological. By the way, let me tell you one more thing. These molecules like to go to the interfaces. So, when I'm washing something, I like to see a lot of bubbles, And you know what it means? The surfactant actually entraps the liquid in air. So your whole detergent really goes up in the air. It's just being wasted. It doesn't go to the dirt. It just goes up into the air as bubbles because liquid is being dispersed in air, okay? In gas. So, but will I ever buy anything that doesn't froth, that doesn't bubble up? No, because it's psychological. I like to think that I'm being cleaned at the time, okay? But this is a fascinating, very interesting field. And you will see that it's involved in a lot of different research areas, but it's also something that we have to be very careful with. Okay. Anything else? Khan or Chai? Yes, I cannot see you, but I'm sure. Oh, okay. Hi.
Yes. Can you hear me now?
Yeah.
Okay. I want to ask first of all, thank you for coming and telling a lot of great things. And like, I want to ask how surfactants can affect the brain chemistry, because like we know that it is used in the drug delivery and those kind of.
Yeah, you're absolutely right. I'm not sure. I don't know exactly what sort of drug delivery it is being used with the brain drugs that are being sent to the brain. If you notice, I use the term targeted drug delivery. So these systems are used in targeted systems, targeted research areas, which means you target the kidney and you send the molecules to the kidney. You see what I mean? Targeted really means you send them to the right point that you want it to go to. So this means if you target it to go to the kidney, you don't expect it to go to the brain. Okay, so this is not supposed to lose its way and be a wild bullet and go somewhere wrong. Okay, that's why I don't know. You have to, as I said, I'm not the expert in the drug delivery systems, but I can imagine how they work and how they are being inserted. You know, some of them are given into the blood. Remember, these drugs do not come alive. They don't go on the market that easily at all. Okay, they take years of research to make sure no damage is done to the other organs, etc. So I don't know if anything to do with brain is being carried out in this manner, but I know cancer drugs are being target delivered to the certain right region. But when we talk about cancer drugs, obviously, as you know, it's not a drug that is used for lung cancer probably is no good for skin cancer or prostate cancer or something else. So obviously, each one is different in structure, each one is different in nature, and each study involves a totally different mechanism. I'm not sure how, these are all being done everywhere in the world, but I've never heard of the brain type of drug delivery systems. But obviously, they're working on specific delivery of specific drugs. So that's why the terminology is targeted drug delivery. It's not just drug delivery. Okay.
So besides the surfactants, like generally how drugs work in brain, like how it can affect, do you know?
No, because the surfactant doesn't do anything. The surface active material, I generalize the surfactants because phospholipids are surfactants as far as I'm concerned, but they're not. You don't call them surfactants, you don't call them detergents, phospholipids, right? You don't say. But anyway, these molecules somehow have to be integrated into the system and have to be pushed out of the body probably after they release their cargo, whatever they're carrying. Do you see what I mean? So that has to be ensured. So it's not that easy. You don't want to target deliver something to somewhere, okay, and then we end up with more damage being done because of the carrier that you sent there. Okay, you want it to release its cargo and then move back and maybe disintegrate, maybe be released from the body in another way. So it's a very complicated mechanism. Okay, I just wanted to introduce you to this world that you will come across vesicles, liposomes as carriers of all sorts of things from now on. You will hear more of them. They're not just in cosmetic chemistry, okay? They're in all sorts of very serious research areas. Do you see what I mean? You just have to, I don't know how the body gets rid of the carrier. We need the cargo, but the encapsulated thing, but how about the encapsulator? The body has to get rid of that. You don't want to keep it inside. That might cause more damage. I don't know. But I'm sure if you get into this field, you'll learn a lot more, and maybe you'll be the next one to talk in this platform and tell people about your findings.
Okay, thank you very much.
Yeah, I just wanted to introduce you to this. I don't know, I find it quite fascinating because they form all sorts of very interesting structures, and they're in everywhere, you know. Okay, this is, any other questions?
Dr. Atay, I have one. First of all, thank you so much for your wonderful speech, accepting to help your alma mater.
Alma mater, yeah.
This was very informative and fun to listen and get all the information from you. I have a little bit of an interesting and hopefully not so complicated question. For the last 5 years, I guess, the economists, ecological economists, have come up with the idea of degrowth. Degrowth in the sense of how to explain it, growing, but growing at the same time not affecting the environment. So I was wondering, like, science is always about progress, and no matter what, as scientists, we will always research and we will always try to get information and like progress and to reach to the beyond of like the human limits. But I was wondering if the term degrowth is possible in terms of science. And what I mean is, of course we're going to research, but would there be a possibility to limit the search to the sense that is not harmful to the environment? I don't know if it will ever be possible, but in a philosophical I was wondering what your opinion is about that.
Okay, I can only give my opinion because, oops, I'm gone.
We can hear you.
We can hear you.
Can you see me? Can you hear me? Okay.
We can hear.
Okay, then I'll talk. I can only give you my. You're absolutely right. I mean, all research somehow do cause damage at the same time, obviously. And we create new stuff, and most of them they're not natural stuff. So we, with the plastics, look at the plastic bottles and the environments and everything. Maybe there will be a consensus on going into certain research areas instead of diversifying, and maybe try to do something collaboratively, I'm going to say, but that seems pretty difficult in this world, right? Not everybody will agree with everybody else. So it's a very, very difficult question. I hope I could answer it, but I really don't know what to say, but you're absolutely right. But I think maybe the best thing to do is to focus on what the world needs. Maybe certain countries could pick up on certain things and give the others, because at the moment it's not balanced at all. You know, we're trying to do our best. We try to save the environment, and you look at some underdeveloped countries, everything you try to do is being is done in the opposite direction in those places. So it's very difficult. I don't know. I wish I could answer your question, and I hope. Can you still hear me?
Yeah. Thank you so much. Thank you.
Okay. Sorry. Yeah, I mean, I really wish I could answer a bit better, but you know, I don't know. I don't know how to solve this world problem. Okay. If you have any ideas, just tell me. Okay.
Thank you very much for your answers and presentation. We're certain that your speech was inspiring for the aspiring scientists in the audience. Professor Atagök, in order to express our gratitude for your speech and help Green Turkish Cause, we will plant 35 new trees via the Tema Foundation on your behalf.
Thank you.
That's great. Thank you. Now that Professor Atagök's speech is over, we will have a 50-minute long break or less. So the break will end at 8 p.m. in Istanbul time and at 1 p.m. in Eastern Standard Time. So that's a 45-minute break. The Zoom meeting will remain open, so you can either stay in the meeting with your camera and microphone turned off, or leave the meeting and rejoin before the end of the break. We will proceed directly with the third student presentation. So Kim Brian Kim from St. Mark's School in Massachusetts should be ready to present after the break. Please prepare your materials in advance.
So bye-bye. Thank you.
Thank you
was
very
very
much.
nice. Thank you. Do stay in touch. Okay. Bye. Thank you. I'm leaving now.
Welcome back, everyone. We will be starting in a minute. In the meantime, we kindly ask our participants to turn on their cameras. Okay, we will now proceed with the fourth student presentation. Kim and Brian Kim will now present their research about the impact of microplastics and accompanied pollutants on the reproduction rate of Caenorhabditis elegans. We once again suggest that our participants write down their questions to ask them at the end.
Hi, everyone. This is Brian Kim, and my research focuses on the impact of microplastics and accompanying pollutants on the reproduction rate of Caenorhabditis elegans. To begin with, microplastics are plastic particles that are less than 5 millimeters in length. Microplastic can be largely categorized into primary and secondary. Primary microplastics are particles that are directly released as small particles. The figure on the right shows some examples of both primary and secondary microplastic. On the other hand, secondary microplastics are particles that deteriorated from larger pieces of plastic. Also, there are several types of plastic: low-density polyethylene, high-density polyethylene, polyvinyl chloride or PVC, and many more. In my experiment, I'll be using LDPE, which is low-density polyethylene, and testing its ability as vectors. Previous studies show that microplastics can act as vectors in transferring chemical additives, pollutants, and antibiotic resistance genes. In addition, these particles can cause intestine damage through organ puncture and long-term retention within the gut. Since the particles are not being digested within the gut, organisms' food consumption rate decreases, which decreases the overall fitness. As said, I'll be testing on C. elegans. These very small transparent nematodes are advantageous as model organisms due to their short reproductive cycle, lifespan, and the fact that they're hermaphrodites, which means that they have both male and female sex organs. This allows self-reproduction, which lasts about 6 to 7 days. They have a lifespan of about 18 to 20 days, allowing short-term experiments. For this experiment, I only used N2 wild-type females, which can reproduce by themselves, to reduce the variability from different genes. Also, I'll be starting all my experiments with L4 stage worms, since that is the last larval stage, and C. elegans start laying eggs during the adult stage. And just for an interesting fact, the Dower stage, as you can see on the bottom left figure, is a stress-free stage that the C. elegans enter when, for example, they don't have any food source. And when they have sufficient resource to grow, then they exit the Dower stage and continue their growth. And I decided to use a terrestrial organism because there has been an abundant research done on marine ecosystem and the impact of microplastic on the marine ecosystem, but compared to the amount of research done on that sector, there hasn't been much evidence and research done on the terrestrial ecosystem. So I thought it'd be important to dive deeper into the less recognized part. I have a total of three experimental groups: control, microplastic, and microplastic plus vegetable oil. For the third group, I soaked low-density polyethylene powder of 500 microns in canola oil for 24 hours and used the filtration process to remove the excess oil. The picture on the bottom left is the filtration process. Since plastic is commonly used for food packaging, vegetable oil is a common pollutant that accompanies plastic. I'll be testing the microplastics' ability as vectors in transferring this common pollutant. As the chart below shows, group 1 did not receive any treatment. Group 2 was treated with 0.005 gram of LDPE and group 3 was treated with 0.005 gram of vegetable oil contaminated LDPE. To test the reproduction rate, I initially prepared 3 replications for each group. During April, I was able to add 2 more samples to each group. Thus, I had 5 samples in total. For the reproduction rate, I started initially with 1 L4 stage worm to make sure no eggs were laid prior to the experiment, and I moved the adult worm, which grew from the L4 worm, to a fresh NGM plate every day. NGM here refers to nematode growth medium, and all plates are treated with E. coli OP50 to feed the C. elegans. After transferring the worm, I used a scalpel to divide the lid into different sections for easier counting, and I counted the number of eggs and newborn worms using a counter. In addition, another dependent variable that can be measured to test the fitness of C. elegans is the survival rate. However, due to the time limit, I was not able to analyze this variable. To measure the survival rate, however, I can start with 5 or more L4 worms and move the worms that survived to a fresh plate every 2 to 3 days. This will allow me to observe which individuals died and record each date for the survival curve. Compared to measuring the reproduction rate, the survival rate is a rather faster process, so increasing the sample size would be easier. I obtained the C. elegans and E. coli OP50 from Carolina and created all other necessary materials at my school's lab. I hypothesized that the group treated with vegetable oil contaminated microplastic will have the lowest reproduction rate, and that the first 2 groups will have a similar reproduction rate. Thus, I used the equation reproduction rate of control or microplastic group minus reproduction rate of microplastic plus vegetable oil group will be greater than 0. The null hypothesis that I had to reject is that the reproduction rate of all 3 groups is equal. As shown in the graph, the control group had the highest reproduction rate overall and during the first 2 days. Although the second and third groups had similar initial reproduction rates during the first 2 days, the microplastic group had a higher average reproduction rate compared to the third group. Before analyzing my data, I assumed that my null hypothesis is true. Thus, the difference in reproduction rate is set to 0, as you can see in the 2 graphs. To analyze my data statistically, I used the t-distribution analysis method. I couldn't use the normal distribution due to the low sample size. The first graph shows the difference in reproduction rate between the first and third groups, which is the control and microplastic plus vegetable oil group, while the second graph shows the difference between the second and third, which is the microplastic and microplastic plus vegetable oil group. This comparison was done to show that the decrease in reproduction rate was not due to solely microplastic, and since the data shows that there is not much difference in microplastic and control group, the significant difference in the reproduction rate of the microplastic plus vegetable oil group proves that the decrease in reproduction rate is not only due to the microplastic itself, but it is actually due to the pollutants that accompanies the microplastic. After calculating the mean and standard deviation of each group, I used my calculator to run the 2-sample t-test. For the first comparison, I obtained the p-value of 0.00348. Now, compared to the initial p-value of 0.068, it significantly decreased. And the initial p-value I'm referring to is the p-value that I obtained when I only used 3 samples in March. Now, p-value in this case represents the probability that the difference in reproduction rate of the 2 groups is greater than or equal to the data I obtained, assuming that the difference is 0. So in this case, there is a 0.348% probability that the difference in reproduction rate of the 2 groups is greater than or equal to the data I obtained, happening just by chance process. Now for the second comparison, I obtained the p-value of 0.00504, which is slightly higher than the first comparison. Again, compared to the initial p-value of 0.075, it significantly decreased. Prior to the data analysis, I chose to use 0.05 for the alpha level, and since both p-values are lower than the alpha level, I can reject the null hypothesis and accept the alternate hypothesis that states that the reproduction rate of C. elegans decreases when treated with vegetable oil contaminated microplastic. My initial data with 3 samples was not statistically significant. Thus, the addition of 2 samples was extremely vital in making my data statistically significant. In the future, I can increase my sample size to 30 to make the distribution normal. I said my sample size is too small to use the normal distribution and have to be extremely careful with the analysis. With more sample size and the relatively similar data trends, I'll be able to decrease the standard deviation and increase the degree of freedom, which will significantly decrease the p-value as a result. Also, as said, I was planning to measure the survival rate of C. elegans. However, I did not have enough time to. So in the future, it would be important to assess the survival rate of C. elegans under vegetable oil contaminated microplastic to expand the analysis of the fitness. Also, it is very important to test different additives or contaminants that are commonly found in plastics, such as heat stabilizers or antioxidants. Finally, I must find an environmentally relevant concentration. Most of the studies I read utilize dry soil or a solution to calculate the concentration of plastic. However, in order to count the individual eggs, I had to culture these C. elegans on agar, making it harder to measure the microplastics concentration. Thus, I had to choose a reasonable concentration so that the worms and eggs were visible in the presence of microplastics, which is why I chose 0.005 grams. Thank you.
Thank you for your presentation. We will now be proceeding with the Q&A session. Participants may ask their questions verbally by raising their hands or send their questions as messages through the Zoom or YouTube chat. Okay, are there any questions? If not, thank you very much.
Well, I have one.
Oh, okay, yeah.
Okay. Thank you, Brian, for your presentation. It was very detailed. I was wondering how long did it take to collect all of the data, to evaluate it? It seems like you put so much effort on this, which is very nice. How long did it take to complete the whole research? And you should have been probably reading a lot of papers before conducting it to get an idea about the use of microplastics in nature, about the general research made in science by the scientists. Did you notice anything anything significant about the use of or microplastics in science, or how can I actually ask it, about the general research that the actual scientists are conducting currently? Anything that you think that was interesting or that you were curious about, if I could explain myself?
So for the first question on the time, so for the reproduction rate, I had to manually count each eggs and the newborn worms. And as you can see on the figure on the top right, these are extremely small worms, and there isn't a figure of the egg, but eggs are even smaller. So I had to manually count them using a microscope. So for each plate, it took about an hour, I guess it took about 40 minutes for each plate every day during the reproductive cycle. And I used 5 plates. So in total, that would be about 200 minutes. And I had 3 replications, so 600 minutes a day, but I divided that into different weeks. So in total, it would be about 3,000 minutes. So yeah, it was really labor intensive, so I didn't really have time to measure the survival rate. And for the second question of anything that I thought was interesting was the fact that microplastic can act as vectors in transferring antibiotic resistance genes. And it was this one article that I read how when earthworms consume these microplastics that are contaminated with antibiotic resistance genes, those genes can duplicate within the gut of the earthworms. And it can spread to different bacteria that can be released as feces, and that can spread even more antibiotic resistance genes around the environment, which can be quite harmful, since more harmful bacteria would be resistant to antibiotics.
Thank you very much.
Thank you. Is that it for the questions?
I don't think there are any other questions. Yeah, thank you for your presentation. We will now be proceeding with the fifth and also the last student presentation. We kindly request that Lauren Chen from St. Mark's School will now present their research titled "A Low-Cost Voice-Controlled Upper Limb Prosthesis." We once again ask that our participants write down their questions for the Q&A session.
All right. Hello, everyone. My name is Lauren Tran, and I'll be talking about the prototype I created of a low-cost voice-controlled upper limb prosthetic solution. Currently, the most common electric option on the market for upper limb amputees are myoelectric prostheses. There's a picture of a myoelectric prosthesis on the left here. Myoelectric prostheses are a functional upper limb prosthesis option that looks anatomically similar to a human hand. They utilize electromyography as the control method, and there's a picture on the right. In this control method, electrodes are placed on the surface of the skin and pick up muscle signals that are then translated into prosthesis functions. However, there are many problems with these market solutions. They are expensive, they require extensive training, and they lack easy customization for resizing, which is especially important for pediatric patients who frequently need their prostheses resized as they grow. Recognizing these challenges, I wanted to create a solution that makes prostheses accessible to a wider demographic. After researching, I realized I could make this happen by utilizing 3D printing, which has recently risen in popularity and become much more widely available. 3D printing provides for relatively low-cost solutions that are easily customizable. Then, as a solution to the extensive training required to reliably use electromyography, I decided to utilize readily available platforms and create an intuitive control method, and I decided on voice control. Here, I've created a high-level systems diagram to better explain the workings of my prototype. Beginning on the bottom left-hand corner, the user speaks into their smartphone, which recognizes the command in Google Assistant. Then, in the cloud, there are two existing platforms, If This Then That, otherwise known as IFT, and Adafruit.io. I created applets in the IFT platform, which takes the command from Google Assistant and translates it into data that then gets sent to the second platform, Adafruit.io. The Adafruit.io platform collects all the data from Google Assistant via IFT and communicates directly with the program that I wrote. My program tells the servo motors what actions correspond to the data from Adafruit.io. And my program is uploaded to the Feather Huzzah microcontroller, which is positioned just to the right of the middle, right here. This microcontroller is located within the forearm portion of my prosthesis and sends signals to the servo motors to open and close the fingers of the hand. Throughout this process, I followed the design thinking engineering cycle methodology during my research and prototyping process. I repeated the define, ideate, prototype, and test stages several times throughout these past months as I modified and bettered my prototype. I have broken down the challenges I encountered into 3 main design cycles. They are software, electrical, and mechanical. First, I have the software design cycle. One major problem I encountered was that my servo motors had a limited range of motion. This meant that my servos would not turn enough to pull the fishing line and close the fingers of the hand completely. I resolved this issue by defining a maximum and minimum value for the servo, which allowed the servos to turn a full 180 degrees rather than the initial 90 degrees. The second issue deals with my intuitive control method. During the testing of my prototype, I created sliders in Adafruit, shown in green on the left. And setting these sliders to different values would set the servo motor to different positions, either closing or opening the hand. However, this design cycle required that the command have a numerical value in it. I decided that having to give commands like open the hand to 20 was not a very intuitive control method. So in the next iteration of my prototype, I created toggle switches in Adafruit, seen in red on the right, that corresponded to a command clearly associated with an action. So this is the point at which I could say open hand or close hand, and the prototype would do as instructed. I found this to be a much more intuitive system. Next is my electrical design cycle. My initial design utilized micro servos because they could be powered directly off the microcontroller and didn't need an external power supply. However, I soon realized that the micro servos didn't have enough torque to pull the fingers close. To resolve this problem, I switched to standard servo motors. There's a picture in the center there, which have about double the amount of torque, but require significantly more power. So another problem arose out of my solution. While the regular servo motors provided adequate torque, they also required more voltage and power. To resolve this new issue, I needed to connect the prototype to an external power supply in order to provide the servos with adequate voltage. The full system, including the bench power supply, is pictured on the right. Finally is my mechanical design cycle. The mechanical issue I encountered involves the servo motors. I only had 2 servo motors that rotated in the same direction available. And when I mounted these servos inside the hand, they were forced to mirror each other, meaning they were not turning in the necessary direction. I devised a software solution to compensate for this mechanical limitation and force the servo motors to rotate together. And you can see them moving together over on the left. Next, I have a demo video, but I think I forgot to share sound, so I'm going to reshare my screen. Okay. Close hand. Closing hand. Hey Google, open hand. Opening hand. Hey Google, pinch. So that's the demo video of the final prototype that I created over these last several months. But there's a couple things that I want to note about the video. I would first there's a clear delay between when I give the command and when the prototype is actuated. I worked hard on shortening this time delay, but it is not something I have a lot of control over because it has a lot to do with the time required for each of the programs to communicate with one another. The second thing I would like to point out is the activation command, hey Google. This was only used for demonstration purposes and I could easily cut it out. Finally, You can see how the fingers each close different amounts. It is most obvious in how the pinky closes much further than the middle finger. There is fishing line that runs through each of the fingers and is connected to the motors located inside the wrist. Each of the fingers requires a different amount of fishing line to be pulled in order to close completely. But because there are multiple fingers connected to each motor, it is very hard to pull each finger the exact amount, so I had to make a couple compromises. On this final slide, I wanted to define areas for improvement and begin the brainstorming process for how I can further improve my project. The first is very important to me. I want to have a portable power supply because the current bench power supply is not a practical solution. No one's going to be carrying it around in order to use the prototype. I did several trials with 9-volt batteries, but they were ultimately unsuccessful. And I realized it's because the battery wasn't putting out enough current, which the bench power supply did. I would also like to add padding inside for fitting and comfort, and grips on the fingers and palm for more practical and functional use. Both of those have a lot to do with how it's going to be secured to an amputee and a user when it comes time to do that. And then finally, it would be amazing to organize patient trials and collect user feedback. Thank you all for your time. I'm happy to answer any questions that you have.
Thank you very much for your presentation. You can ask your questions through raising your hands, or as we said before, you can write your questions in the Zoom or YouTube chat. We have a question in the Zoom chat. I can read it out loud. What programming language did you use to program the hand?
It was all written in Arduino, which is C++. But I use like the Arduino coding platform in order to upload it to the microcontroller.
Hi, Lauren. Thank you so much for your presentation. And the video looked very cool, by the way. I really liked it. I have 2 questions related to this. First of all, how much did the experiment cost? Because you mentioned that your overall aim is to make this possible for people who are in need. So how much does it cost to complete everything for the research? And 2, are you taking computer science electives in class? Which subject is actually helping you? Do you have any design lessons? When did you actually have the time to make all of this experiment? In which lesson did you have time to complete it? Thank you.
So to answer the first question, it's sort of a long, complicated answer. But basically, the myoelectric prostheses on the market range from $5,000 to $75,000. So I had a lot of room to cut it down. The 3D printer that I used is a very specific 3D printer, and the material that it uses is very specific, and you can only buy a certain material to use with the printer. So if you buy the printer and the material all together, that's pretty expensive. One whole package of material is about $300, but you can make probably 7 prototypes out of it. So it would be 300 divided by 7. And then everything else of like the microcontroller, and then there's elastics and fishing wire, that's probably less than $100. So my entire prototype would be less than $300 easily. As for how much time I spent and where did I get the time to do it, I have taken several coding classes before, and that's how I was able to write my program from scratch. There's not a lot out there about how to make voice-controlled prosthetics, so that's something that I had to come up with on my own. But the time that I had to do this is my school runs a program called the STEM Fellowship, and they give us 2 80-minute blocks and a 45-minute block each week to like just time to sit down and do our independent research. So I spent that class time working on it. And then I spent lots of nights and weekends, just any free time that I had to really pull this off. And the hands-on prototyping process was from November to May. So it was over the course of several months, but I had done lots of reading of papers and refreshing my coding skills and everything over the summer leading into it. As for where I learned to design, I didn't actually design the hand itself. I decided that I didn't want to spend so much of my time doing that because my interest and passion is really in the robotic side of things, the coding and the electronics. And learning how to CAD and design a 3D print would take me a really long time. So I got my design off of an open source website called Thingiverse, and I've cited it in my paper and everything. So yeah.
Thank you very much for your participation. So Kanachi has a question.
Okay, hi. Like I want to ask, like, do you have new approaches to develop your product or something like that, or in different fields?
Like how will it be used in different fields?
Mm-hmm.
Well, it's a combination of a bunch of different fields. So biomedical engineering is sort of the overarching field, but since it's a prosthetic, it can be applied to like occupational therapy and physical therapy and different things like that. I'm not sure if that's exactly what you're asking.
Mm-hmm.
Or
like I'm working on actually AI-based speech recognition techniques or something like that. So like I thought maybe that kind of things maybe help to can help for your experiment too, because like different pattern of speech can generate different responses or something.
yeah, so that would actually, that's like a really strong connection to my project is speech recognition versus voice recognition. Mine is really speech recognition. And the difference is the microphone can just recognize when someone's talking, but voice recognition would be differentiating between my voice versus your voice, and mine can't do that. So that's a field that people are working really hard to try to develop, because in reality, if someone has this prototype and I say open hand, then it's going to work even if they don't say it. And that's when it becomes like a serious problem. And that's when we really need the voice recognition part of it and differentiating between different people's voices.
That's a big problem, I guess.
Thank you very much again for your participation and answering all of our questions. All of the research presented by our participating researchers were really breathtaking and informative. We learned so much from all of you.
Okay, it's now time for prizes. Before that, I also would like to thank all the presenting authors for sharing their research, and to the participants for giving their undivided time and attention. It is now time to announce the best paper of the event, which is the paper that has received the most points from our evaluators. And the author is Batu Yalçın, with his paper titled "In Estimating Ground State Energies of Atoms and Molecules." Congratulations, Batu. It was a paper of remarkably high quality, and your presentation was amazing. Thank you very much.
Thank you.
We have more prizes for other participants. These are the Sabancı University High School Summer School Prize, the International Journal of High School Research Prize, the University of Rochester Online Pre-College Program Prize, and the Özyeğin University Summer School Prize.
First, the Sabancı University High School Summer School Prize. The winner of this prize will be able to attend the summer school program with full scholarship.
The first winner author is Batu Yalçın with his paper titled Quantum Computing in Estimating Ground State Energies of Atoms and Molecules.
The second winner is Kerem Berk Bozkurt with his paper titled A Novel Paradox as an Argument Against the von Neumann-Wiener Interpretation.
Our third winner is Alia Kurt with her paper The Science Behind the Constitute of Oblak and an Investigation of the Suspension's Behavior.
Congratulations, Batu, Kerem Berk, and Alia. Our team will be contacting you afterwards about the prize. We will now be continuing with the Özyeğin University High School Summer School Prize. The winner of this prize will receive a 50% scholarship from Özyeğin University's Summer School for high school students.
The first paper to win this prize is titled Alternative Ways to Administer Vaccines During the COVID-19 Pandemic, whose authors are Kerem Demir Karahan and Ali Kaan Nali.
The second paper to win this prize is called The Usage and Consumption of Microplastics, whose authors are Atilla Görgün, Poyraz Okutan, and Mert Demiraydın.
And the third paper to win this prize is titled To What Extent Soil Type Affects CO2 Concentration, Temperature, and pH in Mesocosm, and was written by Aybike Yeşim Eski Bozkurt.
The fourth paper to win this prize is titled To What Extent Immobilization of Yeast in Alginate Beads Affects its Reusability and Stability by Selin Özer.
The fifth paper to win this prize is titled The Effects of UVC Light on Plants and was written by Gülrana Özkaraca, Diyar Kılıç, and Ege Doğan Bakalcı. Congratulations to you all. Again, as a reminder, our team will contact you about the details of the prize.
We will be continuing with the University of Rochester Pre-College Program Prize. The winner of this prize will receive a 50% scholarship as well as direct admission for URochester 2022 virtual pre-college programs. The first person to receive this prize is Batu Yalçın with his paper Quantum Computing in Estimating Ground State Energies of Atoms and Molecules.
Our second winner for this prize is Kerem Berk Bozkurt with his paper titled A Novel Paradox as an Argument Against the von Neumann-Wiener Interpretation.
Our third winner is Kuman Brian Kim with his research titled The Impact of Microplastics and Accompanied Pollutants on the Reproduction Rate of Cyanorhabditis elegans.
Our fourth winner is Punavit Kasan with his research titled Finding the Exact Value of Cosine Pi over 7.
Our fifth winner is Alia Kurt with her paper The Science Behind the Constitute of Oblak and an Investigation of the Suspension's Behavior.
Our sixth winner is Zifan Karl Go with his research titled Applying Transformer Deep Learning Model on Low-Level Program.
Our next winner is Jacqueline Zatrioske with her paper JNK Signaling as a Mediator for Gene Differentiation in Human Umbilical Vein Endothelial Cells exposed to high glucose.
Our eighth winner is Selin Özer with her paper To What Extent Immobilization of Yeast in Alginate Beads Affects its Reusability and Stability.
And our final winner for this award is Alison Bechert with her research titled The Effects of Polyphenol-Rich Diet on Alzheimer's Disease Progression in a Drosophila melanogaster Model. Congratulations to you all.
Now, it's time to announce the winners who will get the opportunity of publishing their research at the International Journal of High School Research. Our winner in the field of physics is Batu Yalçın with his research titled Quantum Computing in Estimating Ground State Energies of Atoms and Molecules.
Our winner in the field of mathematics is Punavit Kasan with his research titled Finding the Exact Value of Cosine of Pi over 7.
Our winner in the field of biology is Kuman Brian Kim with his research titled The Impact of Microplastics and Accompanied Pollutants on the Reproduction Rate of Cyanorhabditis elegans.
Our winner in the field of computer science is Zifan Karl Go with his research titled Applying Transformer Deep Learning Model on Low-Level Programs.
Our winner in the field of chemistry is Aybike Yeşim Eski Bozkurt with her research titled To What Extent Type Affects CO2 Concentration, Temperature, and pH in Mesocosm.
Congratulations to you all. You'll be able to publish your paper in the International Journal of High School Research free of charge. Again, our organization team will be contacting you after the symposium. Moreover, the editorial team of the International Journal of High School Research granted all HiSci participants with a further 10% discount to publish their papers. In case you are interested in claiming your discount, please be in touch with our team. As long as you submit your paper into International Journal of High School Research and they accept it, your 10% discount will be applied.
Having finalized all of our award announcements, we would like to once again clarify that all award arrangements are according to bilateral agreements between HiSci and its partners. Therefore, in the event that a number of authors cannot accommodate an award or the terms and conditions of our partner institutions require a change in the distribution of an award due to reasons such as but not limited to a previous publishing of a winning group's paper, HiSci reserves the right to make changes to its award allocations after the symposium's date. Therefore, please keep an eye out for further emails from our side, since the award allocations are not finalized until all of the winners confirm their enrollment or submission and abide by our partners' individual terms and conditions.
Although only a percentage of our researchers will have the chance to receive an award, we would like to once again state that HiSci is not a competition, but a research event that wants to prepare high school students in the field of academic scientific research. Therefore, we can say that all of our writers could achieve the same by conducting all kinds of wonderful and authentic research. So, we would like to thank all of you and your advisors for such great work. And we will look forward to working with many of you for the creation of HiSci Archive that will store most of our successful research papers to be inspirations for other young researchers.
Speaking of other young researchers, I would like to first thank my fellow founders of the High School Science Symposium, Berkcan Uğurda, Yasemin Şukal, and Aras Karlıdağ, as well as our teammates Nazlı Kırçiçek, Ekin Kahraman, Ezgi Zehra Ovacıklı, Berra Vatansever, Tuna Tarakçı, Andaç Durmaz, Işık Aysel Kıymaç, Zeynep İpek Kayalı, Eda Ergin, Elen Çiçekli, Salim Sinan Tuncer, and Elif İpek Er Yılmaz and Akif Yahya Dirican. I congratulate you all. You have worked very hard and played an indispensable part in bringing this event to life.
Before we end our session, we also would like to extend our thanks to everyone who has helped us through this rocky road. The Dean of Student Activities, the IT Department, the International Development Office, the Robert College Faculty, and finally our beloved and supportive advisor, Dr. Gözde Kamer, who has always been with us since the first moments of our foundation. Thank you so much for never denying your help and assistance. HiSci, this event through which we all put forth so much hope and faith to promote research among our high schooler peers, wouldn't have been possible without your help and contributions.
With that being said, this was the second year of the High School Science Symposium. In a matter of just 2 years, we have created the first of its kind symposium in Turkey and brought our platform to an international level. Thus, we may probably have had some shortcomings in the organization process, but we will always try to improve HiSci with your help by learning from our mistakes. Therefore, we are sorry if there has been even a minor confusion caused by us. We will keep working for a better and more successful HiSci with a new theme, new papers, new ideas, and new initiatives in 2023. We look forward to working with you all in the upcoming years as well.
All of your research and participation were really meaningful to us. Therefore, we would like to end our event by thanking each and every one of you for your great contributions.
Congratulations and a big thank you for all of your hard work and participation. With that being said, HiSci 2022 has now come to an end. We wish you a great summer break and hope to see you again in next year's symposium.
Thank you.